Hello students, am I audible and visible? Let's see, 1 minute. Are we live? Yes, 1 sec. Are we live? Are we live? Yes, we are live. You cannot see me? Yes, we are live. Now all of you can see Gopika ma'am. Yes or no? Yes? Hi, hi. So, good morning students. Welcome to Vedantu NEET English. I am Miss Gopika, your biology educator. So, today we will be doing excretory products and their elimination. Okay, this chapter has a weightage. We are expecting 4 questions from this chapter. That means expecting 16 marks. Okay, 12 to 16 marks. So, we will be doing the entire chapter, detailed lecture, every line of NCERT. I mean, I will not be putting NCERT snips, but then I'll be teaching every single line of NCERT and some additional information which has been repeated in the past year. So, are you all ready? Okay. Yes, I will definitely make it easy. So, those who are new to this channel, quickly subscribe to the channel. At the same time, like. Okay, mark your attendance, all of you. Quickly mark your attendance. Ready? Let's start the class. Hello, I'm good, Sharath. How are you all doing? Let me know in the chat box, how are you all doing? It's been some time that we met. So, how are you all doing? Are you all doing good? How is your, the stress level after the application form is out? How is everything going? Yes. Yes, I am good. Thank you so much. All of you, let me know. I can see a lot of new names. I'm so happy to see you all. Okay, so quickly let me know in the chat box. Yes, okay. Okay, did you all share the link? Did you all share the link with your friends? That though you people didn't do. You won't do that. No, sharing link with friends is the most difficult part. When I ask, you will be like, Ma'am, I don't have friends. Okay. So students, this is the flow of the class. I have put a lot of PYQs in this in the end of the session, in the middle of the session. I have also put some diagram based questions. So you will be solving all of it. So I want all of your attention to be there. If the chats are disturbing, please off the chats. Okay. Only when I ask you a question, you come on the chat and reply. If you feel like there's a lot of disturbing people or a lot of people are putting a lot of messages, please block them and move on. Okay. Because this is a medium where there is a lot of people. Okay. We cannot control. I cannot control the crowd. Yes or no? Okay, fine students. So we will start. So first question is, what is excretion? Right? That is the first question. Yes, okay. Okay, great, fine. Students, so when we talk about excretion, you should also know something about digestion, because digestion and absorption chapter is removed from your syllabus. We also know that you should understand one thing very clearly. In digestion and absorption, you study that the unabsorbed food. What food? Unabsorbed food. Right? What do you call the unabsorbed food as? Fecal matter, right? That unabsorbed food will be going out as the fecal matter from your digestion and absorption chapter. That is all about it, right? You digest the food, whatever is not getting digested or the unabsorbed nutrients or unabsorbed waste will be going out as fecal matter. Now, you'll be like, ma'am, what is the difference between excretion chapter then, right? Yes? Yes, okay. So, students, the topic here is excretion. That means here we are basically talking about elimination or your urine formation, right? Urine formation. Here we are talking about what? We are talking about urine formation or the removal of your liquid waste. What is the liquid waste? Urine, right? So, for this to happen, for what to happen, for excretion to happen, we have some people in tools are there, some tools are there. What are those tools, students? Kidney, 2 bean-shaped kidney, right? Ureter. I want all of you to write the notes with me, okay? I will not share the notes, right? And the urinary bladder, which is a triangular structure. Yes or no? This is the normal excretory, your entire system, right? What is this bean-shaped thing? Kidney, right? This is kidney. What is this 2 tube structure that you're seeing? Ureter. Yes? Yes or no? Are you all with me on this? Yes, yes. Hello, hello. Tell hi to everyone. Okay? Yes. Students, what is it? Ureter. And what is this? Where the urine will be stored will be the urinary bladder. Urinary bladder. Now this, through the urinary bladder, your urine will be expelled out. Now this is all about it. Okay, this is all about it, but you will have to study a lot about it. Okay, I have told this dialogue multiple times. Okay, so students, now all of you let me know in the chat box, what is excretion? Imagine you are telling a person who does not know anything in science. What will you tell? What will you tell? You cannot tell that it is micturation, it is glomerular filtration, it is tubular reabsorption secretion. Use simple words and tell me what is excretion. All of you, come on. In the chat box should move very fast. Okay, those who have already done this chapter with me, it should be like a revision for you. Those who are doing it for the first time, let's break it down. Okay, so tell me, if you're telling your mom or dad or your sister or anything, what will you say excretion as in simple terminologies? Can I say removal of nitrogenous waste? Can I say it is the removal of nitrogenous waste? Yes or no? It is basically removal of nitrogenous waste. Simple words. Now the doubt will be like, what is this nitrogenous waste? Yeah, people can ask that, right? What is this nitrogenous waste? Students, nitrogenous waste is nothing but the waste that is formed by any metabolic activities. Any metabolic activities. So you know our body is formed so complexly to be able to do all the kind of metabolic activities. Yes or no? So whenever we do a metabolic activity, some waste will be emitted out. Those waste are called as nitrogenous waste. Now, such a big organ cannot just be doing excretion. It has to do more things, right? It will also do something called as osmoregulation. All my students know osmoregulation, yes or no? It will also do something called as osmoregulation. Very good, very good. Removal of metabolic waste. Yeah, you can also say removal of metabolic waste instead of nitrogenous waste, because nitrogenous waste is formed from metabolic activities. Okay, very good. Yes. Now, yes students, so who all know what is osmoregulation? Something regulating, right? Something is regulating with the outside temperature, inside temperature. So there's an easy way to remember, I'll tell you. All of you have been in school, all of you have attended assemblies, right? Have you seen in assemblies people fall down? People just like, randomly someone will be standing, they will be telling national anthem or something, and one of your friend will fall, or one of the girl from another class will fall, and you will be like, why are these people fainting? Yes, one reason is they have not had their breakfast, but then one more thing is they are regulating their body nature with attendance, marks card, paper, then maths class, Hindi class, whichever class. Okay, I was very scared of maths class and Hindi class. So I used to think that should I faint today or should I faint tomorrow? Like, we used to think like that, but then it's not possible. Okay, because teachers have, yeah. Most of you have fallen down. Yes or no? Tell me how many of you have fallen down in your class. I mean, in the assembly when you're standing because you didn't have breakfast and then they'll make you stand in hot sun. So, now, what does that mean, students? That means something is getting regulated, something is getting like outside, like you remember that, oh my god, today I'm going to get marks card or today I'm going to get maths paper, so you will tend to fall down. That is called as regulation, right? So now osmoregulation means it's basically salt and water balance. Salt and water balance. That means in your body, the amount of salt, that is your sodium, amount of salt and water has to be balanced. That is called as osmoregulation. We have learned something about osmoregulation in our animal kingdom. A little we have learned about it, right? We learned about flame cells, protonephridia, Malpighian tubules, all this. Yes or no? Yes. Yes, all of you would have wished to fall, but sometimes, your body will be like, shut up and stand. What are you trying to fall? Right? Even my body would have told, many times it has told me, when I try like, okay, I should fall like this. But then body is not ready to fall. Body is like, no, what if you hit your nose? Okay, what if you hit your head? So it does not happen, okay? Every time it does not happen, but for some people, it naturally happens, okay? It naturally happens. Yes or no? Okay. Yes, very good. Flame cells, correct. Okay. Now share the link with all of your friends quickly. Share the link. Now students, I told you nitrogenous waste. Now we are talking as if we don't know anything about excretion. Okay, we are not even NEET students. We are just basic people who are like trying to learn science. Now I need to know what are the nitrogenous waste. So nitrogenous waste is of three types. Okay, three types of nitrogenous waste is mentioned in your NCERT. There are a lot more of nitrogenous waste. Okay, so the nitrogenous waste are what? Ammonia, urea, uric acid. Ammonia, urea, uric acid. These three are the types of nitrogenous waste. Okay, you will be studying in detail about these three people, and you will also understand why this person is not there in our body, this person is there, why we are not secreting this person. All this you will understand. Is this so far clear? Yes. Uric acid, urea. Yes, even maths class, yeah, I understand students. That's the reason why we have all taken biology. You know, at one point we are scared of maths. It's like maths will eat us up alive, so let's just escape from it. Okay, now this much is clear. So what and all did we do? Unabsorbed food, because students, lot of the students when they write excretion, no, they write removal of unabsorbed food or removal of fecal matter. This is a big confusion. Now that digestion and absorption is removed, then all the students will be in an assumption that excretion, urine formation and fecal matter, everything is, you know, mixed. So that's why I'm telling unabsorbed food is removed with the help of your digestive system, right? It is in your digestive system. Here what are we talking about? In excretion, we are only focusing about the urine formation and expelling of the urine, okay? And this is your excretory system, right? This is the entire what you are going to study today. You will study the anatomy, you will study histology, everything you will study about these systems. So it is the excretory system. So excretory system has two bean-shaped kidney, a ureter, long tube, two tubes forming or joining into the urinary bladder, and here you will have urethra through which the urine will be expelled out. Now such a big organ will not just do excretion, it will also do something called as osmoregulation, basically salt and water balance. Okay. Now simple words, excretion means what? Removal of nitrogenous waste. Now what, from where does nitrogenous waste formation happens? From metabolic activities. Okay. Now what are the three types of nitrogenous waste that you have to study according to NCERT? Ammonia, urea and uric acid. Done. Yes, very good, very good. Okay. So much is clear. Okay. Did you all mark the attendance? I cannot see. I can see 40 people and I can only see 34 likes. Who are the 6 kanjus sitting in the class? Bunking class. We should call them like that. Backbenchers. Okay, come on. Smash the like button. Okay. Now students, we are going to learn osmoregulation. Okay, we are going to learn osmoregulation with the help of a graph. Come on, let's do it. Oh, did it become straight line? Yes. Okay. Yes. Can you all see this? Okay. Yes. Students, what have I drawn? X and Y axis. No, that's what I've drawn. X and Y axis. Now, so we are going to label it. Okay, we are going to label it. So we are going to check osmolarity of the external conditions, okay, and here we are going to check the osmolarity of the internal conditions. Yes or no? What do you mean by internal and external medium? Basically, your body versus the environment. That's all, right? External will be the environment, internal will be our body. Will I be able to regulate if it's hot outside, or if the salt water balance, can I regulate with the outside temperature? These things are the internal and external factor. Now we know what is osmoregulation. We know osmoregulation is basically meaning salt and water balance, right? It is basically salt and water balance. This much is clear. Now students, we have to draw a graph. Okay. Now there are 3 types of organisms. Very carefully listen. There are 3 types of organisms. In this 3 type of organisms, one set of organisms are the ones who is like, I don't want to take any stress. If the outside temperature is changing, if the outside conditions are changing, I am also going to change. I will just modulate or change myself according to the outside temperature. Okay, they are called as osmoconformers. What are they called as? Osmoconformers. Is this clear? Okay. They will have a straight line. Why, students, they will have a straight line? Because they are like, okay, I do not want to take any effort. I will just regulate my temperature depending on the outside temperature. Simple, no? So, this are called as osmoconformers. Okay, these are osmoconformers. Clear? Okay, very good. Osmoconformers. Now, osmoconformers are organisms which will change their temperature or adapt to the osmoregulation with the external temperature. Yes. Now, students, osmoconformers examples. Now, we know that we cannot do this, right? Who will do this? Invertebrates will do it. Okay. Smaller organisms. Invertebrates will do, as well as mycene. Okay. Mycene will do this. Even though mycene is a vertebrate, it is a very primitive vertebrate. So, mycene and invertebrates will do osmoconformers. Now, the question can come in NEET. Mycene, what does it do? It is a vertebrate. Does it do osmoregulation or does it do osmoconform? Is it osmoconformers or is it osmoregulators? So, your answer should be, it is osmoconformers. That means they change their temperature or change their salt and water balance depending on the external medium. These people are stress free, okay? They are like, okay, everything outside, no one is studying, I will also not study. Outside, my friends are playing, I will also play. They are this kind of people. Then we have next set of people who are, straight line. This is not straight line, no? Yes. Okay, students, these are osmoregulators. What are they? Osmoregulators. Students, can you see all the colors? They are osmoregulators. Is the colors visible? Yes. Okay. Did you understand or not? Students, osmoconformer simple. That means outside whatever is happening, I will modulate my inside. Okay, like that. That's all. Like students, how we have warm bladder, like cold bladder, right? Polyokilotherms. So what do? They will basically change, right? Outside temperature, whatever is happening, they will regulate the inside temperature. We cannot do that. Why? Because we have a constant temperature. That is your 37 degrees Celsius is my body temperature. Whatever is the outside temperature, I will not be able to regulate. Same way, here we are talking about salt and water balance. So, osmoconformers are the ones who will change depending on the external medium. Is this clear? I think Anish Pandey asked. Yes? Okay, great. Okay, now osmoregulators. Osmoregulators are who? That means they will stay constant. Whatever is happening outside, they are not bothered. They will have a very constant temperature like us, right? A very constant salt water balance. So, osmoregulators are the ones that do not change, okay? They does not change with the external temperatures or external medium. External medium. Who will be this? Obviously, it will be vertebrates, right? All the vertebrates, okay, they will be, examples are all the vertebrates. All the vertebrates except mycene, okay? All the vertebrates except mycene. Is this clear so far? Yes? Students, look here. This is osmolarity of internal medium. Here it is osmolarity of the external medium. Okay. Is this clear, Rishabh? Yes, very good. Vertebrates. All the vertebrates will fall under osmoregulators, which does not change with the external medium. Whatever happens outside, I am not bothered. I have a job here. I have come for that. I will do that. Okay. That is this set of people. Now, students, we have one more set of people, which are the most funniest set of people. I'll tell you why. They have a very fancy life by themselves. Students, what do they do, no? Say we will draw from here. Wait, 1 second. Okay. Can you see this? Can you all see this? 1 second, add. Is this so much clear? 1 second. Yes. So students, look here. These are partial osmoconformers. That means they will be like, okay, let me do one thing. Okay, let me do one thing. I will start regulating my temperature depending on the outside temperature. In the starting, they'll have that energy. Okay, I will do this. Okay, at one point, they will be like, no, I can't do this. So they'll just become like, okay, whatever is happening in the external, I'm going to change. So at one point, they'll be like, okay, I'm going to utilize the ATP and I will regulate. I will maintain my body temperature. Okay, but after some point, they'll be like, no, it's not possible by me. So what I will do, whatever is happening external, I will also just adapt to it. They are called as partial osmoconformers. Okay. Yes? Yes. Partial osmoconformers. Very good. Okay. Partial osmoconformers. Don't see the graph and get confused. Just understand this people starting they would be trying to do osmoregularity, but then later they'll realize I do not have the energy to do that. So I will just change my temperature depending on the outside temperature. So they are called as partial osmoconformers. Now examples are elasmobranchs. Okay, elasmobranchs. Students, elasmobranchs are your sharks and rays. Okay, sharks and rays. Those are the example. You can remember that way only, or you can remember as elasmobranchs. Sharks and rays. Okay. Understood? Now, these all come under osmoregulation. Okay, do not get it confused. They are all doing they are all doing salt water balance, but they have chosen their own methods. Do I have to do depending on the external temperature, or do I want to keep changing according to the external temperature, or I want to be constant? That's all. Okay, we also change our temperature. Like, see, we also adapt, right? If it's too cold, we wear overcoat. If it's too hot, we switch on the fan. All this is our kind of adaptation, but there are organisms which will just keep changing depending on the external temperature. Okay. Yeah. See, Bhuwana, we call organs of invertebrates osmoregulation because they are doing osmoregulation. No, they're still doing salt and water balance. That's what I was trying to say. It's just that they're doing in different ways, right? They're just doing in different way. Yes, elasmobranchs. Clear? Okay, great. So, students, look here. What are all examples? This is very important. Mycene. Okay, mycene is an separate, who tell? Hackfish, completely separate from your vertebrates. Even though it falls under vertebrates, it's a very primitive vertebrate. That's the reason this falls under osmoconformers. Yes? Clear? Okay, great, fine. Shall we continue? Shall we continue? Yes, okay. Students, first topic is your nitrogenous waste, okay, which is your nitrogenous waste, and what do they do, right? So, first we have ammonia. Whom do we have? Ammonia. So, students, ammonia is not kept in our body for a longer time because it is very, very toxic, okay. So, among your ammonia, urea and uric acid, ammonia is the most toxic, okay. It is the most toxic one. Okay, it is the most toxic one. And why it is not kept in our body for a longer time? First reason is this needs a lot of water, right? It needs a lot of water to be removed. Okay, so that's the reason we cannot afford to give. See, we do not consume, or we do not sleep, or our habitat is not in water that we can, take in so much of water to remove our ammonia. That is not possible by us. So that's the reason it requires a lot, okay? It requires a lot of water for removal. Is this clear to all of you? Easy, students, do not make it complicated. Keep it very easy. Keep it very easy. Okay? Yes, very good. Highly toxic, and they need a lot of water to be removed out. Now students, I told you we cannot keep it in our body, right? We cannot keep it in our body. We cannot afford to wait until kidney comes and slowly filters this out. The blood will come, filter. That much time we don't have. We will only we will vanish. So that's the reason it has to be readily removed. Okay, it has to be readily removed. Is my handwriting clear on the board? Okay, it has to be readily removed. Now, if it has to be readily removed, I already told you that I cannot wait for kidney to come and do the job. So, what will I do? I will do another thing, that is, we will try to remove the ammonia with the help of diffusion. That means from the body surface. So, ammonia will be removed. Okay, ammonia will be removed from body surface with the help of what? With the help of diffusion. So, how do you connect all this, students? How do you remember all these points? First thing you should understand, it is very toxic. It needs a lot of water. We do not have that much of water. So, this is not concerning us. Okay. And because it is very toxic, can you keep it in the body for a longer time? You cannot keep it in the body for a longer time. So, you have to remove immediately. Now, how do you see? I do not have the time to wait for the kidney to come and do this job. So, what I will do? I will just readily remove it from the body surface with the help of diffusion. Is this clear? Okay, easy? Easy? Okay, thank you. Okay, students, that much is clear. Now, because I told you I am removing it from body surface, I can say there is no role of kidney, right? No role of kidney. All of you, please write notes with me. Do not wait for my notes. Your short notes are going to be such a blessing in the month of April. You will be like, yes, this is so good that I wrote the notes. I'm telling you, trust me on this. Your notes will work magic more than any teacher's note. Okay, any teacher's note, your own notes will work magic. Because sometimes students, even I have noticed, even if I see 100 lectures or if I see many videos and just take notes from anyone also, I will not understand until I write it down. Okay, so please do write it with me. It is not going to be that of a task. Okay, and all of you hit the like button. Yeah, very sad, very poor attendance on a Sunday. I don't know where all of them went. Wait. One second. Yes, done. Okay. Yes, students, come on. Yes, I will be doing vision 720. I'll be doing revision for your boards. That time I'll take principles of inheritance and evolution. Okay. Again, exam is on March 19th and 18th. No, so we will do it by then. Okay. Because if I do now revision again, you will forget everything, and again we'll have to do one more revision. Okay, students. So this will be removed in the form of NH4 plus ions. It will be removed in the form of ammonium ions. That's all. Okay. It will be removed in the form of ammonium ions. Now, the question comes, who are this organism? Who are this organisms who is capable of doing or removing ammonia from their body? Those organisms are called as ammonotelic organisms. So the organisms which will remove ammonia are called as ammonotelic, and the process by which they remove is called as ammonotelism. Okay, ammonotelism. Is this clear? Easy, no? Process and organism. Okay, process and organism. Now, I already mentioned to all of you that this ammonia requires a lot of water. Now, can any of you guess and tell me which will be the organism who will be able to give out ammonia? Okay, it has to be an organism that has like their habitat is in aquatic form. Yes or no? Agreed? Okay, so which will be the organism which can do this? I will write some of the examples. Rest of the examples you should tell me. So the examples will be sponges. Okay, your porifera because they are in water. Your cnidarians also can come. Cnidarians, some of the bony fishes. Some more examples can all of you give me in the chat box? Can some of you give me some of the examples in the chat box? Come on. Yes, very good, very good. Aquatic organisms, amphibians. Sharath is sending emoji. Sharath, I have to understand the emoji and write a. Which have you put emoji? Okay, yes, amphibians, correct. Aquatic, amphibians. Okay, aquatic, amphibians. So many examples are there. You can write sponges, cnidarians, some flatworms, some bony fishes, some aquatic amphibians. You can write examples of anything which is in water, right? In water. Is this clear? Yes. Yes. Oh, Gayatri. Gayatri, I had already told it in the class, dear. How to remember the bones of your legs. I have already made a thing in the class. So please watch that video, locomotion and movement, okay? Because, yes. Where's Baswarath, sir? Oh, hi, sir. Yes, bony fishes. Very good. Amazing. Okay, now, students, okay, we will do it in the urea thing. Okay, we will do it in the urea thing. I wanted to tell you one information. Okay, now students, next excretory product is urea. Now, do we form urea? Do we excrete out urea? Yes, we do excrete out urea. Because it is less toxic. Whom are we comparing with? We are comparing with ammonia. Okay, it is less toxic and it does not require a lot of water. Again, with comparison to ammonia, it does not require a large amount of water. Students, it is given very messed up in your NCERT, so please write it down this way only, because in NCERT, top they have mentioned ammonia, down they are mentioning ammonotelic, then somewhere they are mentioning uricotelic. So it is little khichdi form, so we will write in a better way, okay? Yeah. Okay, no, how are you? CM Pandu is hand. Leg also I have told, yeah, leg also I have told in the class. I have not made a video out of it. I have told in the class because it's a very funny thing which I only made. It is not existing. So that's the reason I didn't want to make a video out of it. Okay, yes, students, does not require a lot of water. Now, sometimes in our body also ammonia will be formed. What did I say? Sometimes in our body also ammonia will be formed. So this ammonia, okay, this ammonia will be sent to who? Sent to the liver. Because liver is capable of doing your ornithine cycle or your urea cycle. Right? Liver is able to do ornithine cycle or your urea cycle. So this, what happens, students? We know that ammonia is very toxic. Now imagine our body starts forming ammonia. What will happen? It will become toxic to us. It will cause us a lot of problems. Now we have to avoid it, right? So whenever ammonia is formed in our body, it will be sent to the liver. In the liver, urea cycle or your ornithine cycle will be formed. That cycle will convert this ammonia into urea. Okay? That cycle will convert this ammonia into urea. Is this clear to all of you? So the question can come in such a way that, do our body form ammonia, and if yes, then how is it, you know, converted to urea or anything like what if it is too toxic? So all this can come. Okay, all this can come. So your answer should be that. Okay. What is happening? All are telling vanakkam to each other. Yes. Okay. So students, is this clear? So it is less soluble in water. Okay. Less soluble in water. Come on. Okay. Student, by the end of this session, can we make it 200 likes? Yes or no? All those who join newly, smash the like button. Come on, all of you. Yes, students, so it is less soluble in water. It requires very less amount of water. Now, the question comes is, we excrete out urea. So, what are the other examples? Okay, other examples. So, before we go into the examples, now we need to know if an organism is giving out urea. What do you call that organism as? You call it uricotelic organism. And if the process by which this comes out, what is it called? Urecotelism. Okay, ureotelism. Okay, look at the spelling, students, because ureco and ureo you might get confused. It's like Oreo. Okay, ureotelism, ureotelic. Okay, ureotelism and ureotelic. Now the example. Is, first of all, mammals. We come into place. Yes. Who are the other examples? Students, remember when we learned frog, I called it uricotelic organism. So that means adult amphibians. Adult amphibian. Yes or no? Got it? Adult amphibian. Then there are a lot of it. Some fishes, cartilaginous fishes. Yes. Okay. Yes. Is this clear? Urecotelism. Yes. Fine. Mammals, adult amphibians, some of the cartilaginous fishes. Okay. Question to all of you. When ammonia is formed in our body, right? How is it removed out? How is it removed out? All of you tell me in the chat box. When ammonia is formed in our body, how is it removed out? Readily removed out in our body. Chalo, batao. Come on, let's make it 100 likes. Share the link. Laharika, share the link with your friends. I know all these people have a lot of friends, yeah, but then they're very shy. They're like, ma'am, I cannot. I can only send her reels. I cannot send her class. Students, I want the process name. If ammonia is formed in the body, how will it be removed? Yes, very good, Sharath. Very good. Very good. Yes, all of you are telling kidney, I understand. All of you are telling kidney, I totally understand, students. But I told you one thing. I told you about one cycle, right? So that means what is happening? Yes, ultimately, kidney will remove. But the thing is, ammonia will go to the liver. From liver, it will get converted to urea. Okay, so you should say, with the help of ornithine cycle, it will be removed out through the kidney. Is this clear? Yes. Hi, Vijayan. Yes, very good. By converting into urea. Yes. Clear? Very good, very good. Yes, Rupa. So, students, mammals, adult amphibians, cartilaginous fishes. So, this way you will have to tell that ammonia which is formed in our body will be converted with the help of ornithine cycle or urea cycle in the liver to urea. Okay, ultimately, okay, whatever happens, kidney will remove it, no problem. But this much you have to remember. Clear? Fine. Done? Did we do everything? Yes, okay. Now, one more question I have. Students, do you think entire urea will be removed from our body? Entire urea will be removed from our body? Yes or no question? Tell me, ma'am, will entire urea be removed from our body? Come on. All of you tell me in the chat box. Bhuwana, the lecture might go for, it's a long chapter. So, sometimes 4 hours, sometimes 5 hours. Okay, it also depends on how fast you people are answering also. Yes. No, ma'am, completely urea will not be removed from our body. Very good. Students, so understand, okay, some of the urea will be maintained, okay, and a PYQ question, please make a note, I am putting a star here. Some of the urea will be maintained in our body for osmoregulation. Osmoregulation. So, you will understand this when I teach you mechanism. Now, you will not understand this. You will only understand this when I teach you mechanism. So, just remember this much, that complete urea will not be removed from our body. Some of the urea will be kept in our body for osmoregulation. Okay, for osmoregulation. And students, please understand, when oxygenated blood is coming inside, okay, when oxygenated blood is coming inside, that oxygenated blood will have some urea in it. Okay, so see, oxygenated, deoxygenated is basically spoke in the terms of presence of oxygen, purified, all this. But when you talk in the case of kidney and everything, in the oxygenated blood also some amount of urea will be there. Okay, that urea will be removed out in the kidney. Is this clear to all of you? Do not get yourself confused. Okay. Yes, very good, very good. Osmoregulation, some amount is left to maintain osmolarity. Okay, very good. Yes, amazing. Okay, so students, oxygenated, deoxygenated, understood. Just understand this, that the blood which is coming into the kidney with the help of renal artery, no, I will draw that and show that blood will have some amount of urea. That urea will be removed out in the kidney, but some amount of urea will be maintained in the body for osmoregulation. It is for one of the mechanisms to work. Okay, students, the chapter is very easy. I will make it as easy as possible, but there are some concepts that you need to, try to connect and learn. Try to connect and learn. Okay, fine. Next, let's go to uric acid. Uric acid is the least toxic. Then you will be like, ma'am, why don't we give out uric acid? Students, we are not giving out uric acid because this uric acid is a semi-solid waste, first of all, and secondly, it's a very expensive process. Expensive process matlab, it's not like I have to pay a lot of money or I have to buy it from Amazon, Swiggy or Zomato. It means that lot of ATP will be utilized during this formation. Okay, lot of ATP is utilized. So because of that, this is a very, very expensive process and we hardly have energy for ourselves to stand and teach like this. Upon that, if we have to give out so much uric acid and energy, we will die. Yes or no? Yes or no? Okay. Yes, it requires very less amount of water. Okay, requires very less amount of water. It is semi-solid or we call it pellet form. Okay. Now we have something to remember. How will you remember uric acid? You will remember a crow. A crow which keeps targeting you whenever you go outside. It will correctly be like, yes, this is the time for me to do excretion and I will do it on this person because this fellow's or this girl's head looks like a playground. It looks very neat and clean. So I will make sure that wherever this person is going, they get embarrassed. Yes or no? How many of you have had this point here? I have already told my story. Okay. Yes or no? Students, tell me. Yes, exactly. Head is the target. It depends on whose head is that day. Okay, some people tell it is very lucky that if you're going out somewhere and then, a bird decides to excrete the pellets on our head, some of the people tell, oh my God, it is so good. Money will come for you. Blessings will come for you. But nothing will come, only embarrassment will come wherever you go. Okay? Yes? Checkpoint. Yes. So students, crow will decide. Okay? Crow will decide who I should target, when I should target, who is the museum's piece who is walking around very stylishly. Depending on that, the crow or say any bird for that matter, especially the crow. Okay, crow is very, very in that way. Have you ever washed your car and put outside? You wash your car just 5 minutes, like as soon as you wash your car and you go inside, crow will be like, yes, this is the cleanest surface I have seen. This fellow has washed his car after 1 month. Now it's the right time for me to excrete. So it will put pellet correctly on the washed car. It will never put pellet on the dirtiest car. Okay, so crow has too much hygiene, students. It will not do it on a dirty head, on a dirty car. Okay, yes, so this is how you remember it. It's a pellet form. Have you seen crows, right? Excreta, you would have seen it. It's semi-solid. Okay, it is semi-solid. Yeah, you people are very disgusting. They're fully detailed. They're going in detail about the fragrance, about the color. My gosh. Look at them. They're so much fun. Okay. So much fun they are having. Full in detail they are going. Lecture. Forget about the lecture, no? Okay, fine. They are having fun. Okay, students, so least toxic, it is very, very expensive process. This is the reason why we are not excreting uric acid, because even though it requires only less water, the problem is that it requires a lot of energy. Okay, it requires a lot of energy. Okay, yes, clear? And it is also very important, least soluble in water. Least soluble in water. Clear? Okay. Fine. Now students, which are the organisms which emit this out, right? Whichever gives uric acid will be called as uricotelic. What will it be called as? Uricotelic. Okay, look at the spelling always. And the process by which it will come out is what? Uricotelism. Uricotelism. Okay, clear? Now, examples. All of you pray for 1 minute for your crow. All the aves, right? All the aves. Some insects also, right? Some insects also. Most of the reptiles. Okay? Most of the reptiles. So, most reptiles, aves, snakes also, right? Yes. Clear? Uricotelism. Very good. Very good. Yes. Fine? Yes. Students, one important thing here also is that we also form some amount of uric acid. You'll be like, what? Okay, so students, we also form some amount of uric acid. It is because of the metabolism of purines. Okay, it is because we form some amount of uric acid because of the metabolism of purines. Now, those who do not know purines, it is in molecular basis of inheritance. I don't think any of my future doctors will not know purines. Purines, you remember, right? Adenine, at Gopika ma'am's class, adenine, guanine, you have cytosine, thymine, and uracil, you have pyrimidines and purines. So, out of this metabolism of purines, right, that will lead to some amount of uric acid in our body. Some amount, okay, not a lot, some amount of uric acid. Clear or not? Okay? Yes. Fine. Got it? Very good. Okay. Yes. Clear? Yes. Pure as gold. Very good. Pure as gold. Fine. Okay, students. So, this much is clear. So, we have understood all the types of nitrogenous waste and we have also understood their characteristics better than your NCERT. Okay, NCERT thoda khichdi hai, but if we have categorized it and wrote it. Okay. Now, students, we have to go to animal kingdom. So, there is a big paragraph mentioned here. I will just underline what is required for you. So, you also please underline in your textbook. Okay. Students, first one is your platyhelminthes. Platyhelminthes or your flatworm, planaria, rotifer, some annelids, cephalochordates, amphiaxis, all of these people, excretion, the excretory, what they use is protonephridia. Where is it? Yes. Protonephridia or your flame cells, okay? Protonephridia or your flame cells. This will help in both excretion as well as for your osmoregulation, okay? This will help in excretion as well as for your osmoregulation. So, platyhelminthes, protonephridia, okay? Annelids, flame cells, okay? Then, next one, students, here, which one we'll have next one? Your earthworm. So, all the earthworm here, earthworm category will go for nephridia. Okay, earthworm, nephridia. I'm marking it with same color so you will understand. Okay, yes. Then students, next we have your insects or your cockroach. Cockroach, when we studied, what did we study? We studied Malpighian tubules because they're developing, no? They've come into, like from primitive, they've come into a little more modified. So, we have Malpighian tubules for your insects, right? Malpighian tubules are the excretory structure in most of the insects. Okay, only this much you have to know. Whatever I am marking, that's all you have to know. You do not have to know anything extra. Okay. Yes, then you have antenal cells or your green glands, which are for your prawn. Okay, all the crustaceans. So, prawn can come, crayfish can come, crawfish can come, all this like prawn. Only prawn is mentioned, but in PYQ, they can ask you any of the crustaceans. So, please make a note, understand. You can say antennal cells or you can say green glands, whichever you are able to remember at that point of time. Antennal glands or green glands, prawns is the example. Is this clear? Okay, very good, very good. Okay, done. Can we go? Students, full trailer is over. Now picture is coming. Okay, trailer is over. Now actual picture is coming. Okay, now let's draw. All of you take color pencil, pen, everything and start drawing with me. Start drawing with me. Okay, so human excretory system. So, we know what and all is there in our excretory system, but let's draw it once again. Let's draw it once again. Okay, all of you draw with me. didn't. Yes, drawing with me. These are your ureters. Where will they open into? They'll open into the urinary bladder, right? And then on top of the kidney, we have cap-like structure called your adrenal gland. Okay, and then I'll take another red. Yes, so this much is clear. Then students, we have some right, we have to draw your veins and arteries also, yes or no? Okay. Students, 3D won't come, so we will do 2D. Okay, is this visible? Fine. One second, I'll just. Yes, all of you please draw with me. Okay, all of you please draw with me. So it will be easy for you to understand what you are writing. Yes. Yes, clear? Yeah, structure looks fine. Now students, let's start labeling. So we know about the kidney, we know about the ureter, we know about the urinary bladder. Any doubts in this? No, right? So now only we have to know about the veins. Okay, one second, let me open the charts. Yes, okay, clear students? So students, look here. We have first, we will start with the kidney, right? We will start with the kidney. So this is your bean-shaped kidney. So what and all do you know about the kidney? You know that it is a bean-shaped structure. Yes, it is a bean-shaped structure. And it is, so first we will write it's a bean-shaped structure. It is reddish in color, reddish brown in color. Okay. Reddish brown in color. Yes, this is general thing. It looks like a rajma, right? It is bean shaped, reddish brown in color. And on top of the kidney, you have a cap-like structure which is called your adrenal gland. Okay, adrenal gland. Then you have two tubes which are extending down towards your urinary bladder, which is called your ureters. Okay, and then you have a storage organ which is called your urinary bladder, where the urine will be stored after formation. And at the end of the urinary bladder, you have a small opening which is called the urethra. Okay, small opening which you call the urethra. And on this urethra, there are two special muscles called as your sphincter muscles. Okay, you have two muscles called as your sphincter muscle, one internal and one external. Okay, now students, important part, about your veins. Okay, so you have your renal vein, and your renal artery. Renal artery. So I told you this renal artery carries your oxygenated blood, but this oxygenated blood will have some amount of urea. So it is not completely, it is oxygenated, but it is still toxic. Is this clear to all of you? This is one point I want all of you to remember. Students, it is toxic. Yes? Yes, very good. Okay, renal vein and renal artery. So renal artery is carrying oxygenated blood. No confusion in that. But in that oxygenated blood, some amount of, urea. Some amount of urea is there, because of which the blood is not completely, it is still having some toxic, right? It is still toxic. Okay. So once it goes to the kidney, it will get purified. Okay, this much clear? Okay, great. Okay. Students, now I want to tell you about a very, you will study this in your, when you do MBBS, when you do embryology of your kidney, you will study this. But I will just tell you very very outside thing about this. Students, understand there is two spaces in our body. What did I say? There are two spaces in our body. Okay. Now let's draw a circle. This is a little extra information because I feel like if you don't understand this, it'll be difficult for you to understand one other topic. Okay. Look here, all of you. Okay. So students, our body has two space or like two cavities. Okay. Two cavities. One is called as your intraperitoneal cavity. Peritoneal cavity. Yeah. Yes, intraperitoneal and retroperitoneal. Okay. So students, this is the two spaces in our body or two cavities in our body which will be separated with the help of a peritoneum. Okay, with a layer called as, with a, separation, like how you have septum, no, like that one layer will be there, which is called the peritoneum. Okay, so understand, there will be some organs in the retroperitoneal space, there will be some organs in the intraperitoneal space. Okay, so in retroperitoneal space, what and all will be there? Your adrenal gland will be there, kidney will be there, duodenum will be there, colon will be there, then both the vena cavas will be there. So many organs are there. Understand, your kidney is in the retroperitoneal space. Okay, that is in the abdomen. Okay, so there is a term which is mentioned in your textbook which is called as kidney is a retroperitoneal organ. So whichever organ that belongs to this space or this cavity in your body is called as retroperitoneal organ or retroperitoneal. It is present in the retroperitoneal cavity. Is this clear to all of you? This is a very complicated, like it is by itself a topic in your MBBS, but just understand that there are some organs in retroperitoneal space and in your intraperitoneal space which is separated with the help of a peritoneum. Okay, now in that we have your kidney, adrenal gland, some part of your duodenum, colon, rectum, all this is there. Okay, a lot of organs are there. SAD, PU, PLU, C, KER. Around 9 organs are there. Out of that, kidney is one among them. Is this clear? Okay, is this clear? Yes, okay. Okay, clear students? Fine. Okay, retroperitoneal, that is what it is meaning. Okay, because if you by heart, no, there are chances that you will not be able to write it in the exam thinking that what is this retroperitoneal, because it's not mentioned properly. So kidney is a retroperitoneal organ. Okay, retroperitoneal organ. That means it's basically present in that cavity. Okay, it's present in the cavity. If you want to know more about this, you can ask me. I can explain you some other class. Okay, as an additional information. Okay, but as of now, I hope this is clear. Okay, fine, great. Now, students, other than that, one more thing is that which is just mentioned as a word. So I thought I will explain that also. Students, that is basically about your metanephric, mesonephric, and pronephric. So I'll give you a very small understanding about it. Understand, your kidney development or your all the organs in the body development happens in a very slow stage. Okay, so in the first, like that means the before giving birth, right, before giving birth or during your first trimester, the kidney will be developed as small tubules or small cells. It will not be completely developed. Okay, that stage is done, or that part of the kidney's job is done by pronephrons. Okay, so there is meso and meta. Meso and meta. Okay, so pro will be in the initial stage. That means your first trimester, when the baby is, in the developing stage, is pronephrons will form some cells. Okay, but this will degenerate and go off completely. Okay, it will become non-functional. Then meso and meta. So mesonephronic will again form small tubules and capillaries which will give rise to your glomerulus and your renal tubule. It will be like your renal tubule, but it will not be completely developed. Then the actual kidney, right, this complicated kidney will be formed by your metanephrons. That is why it is mentioned in your textbook that your kidney is metanephric in nature. That means it is made up of metanephrons. Just understand, during the growth of the baby, It starts from pronephron, mesonephron, metanephron. Pro is in a very primitive stage, and then meso will still, like, capillaries will be formed, renal tubules will be formed, but not like complete PCT, loop of Henle, DCT, nothing like that. Just a tubular structure will be formed. Actual kidney, that means the complex structure of your PCT, loop of Henle, everything will be formed by your metanephrons. This also I wanted to mention you. Again, this is again completely one whole anatomy is there about this. Whole embryology is there about it. As of now, just understand this much. Is this also clear to all of you? Is this also clear to all of you? Okay, there are so many words, students. Some caps will be there, blastema will be there, okay, all that. Is this clear? Can all of you tell me in the chat box? Clear or not? Come on, why is the chat's not moving? Yes. Okay, so metanephric in nature. Metanephric, okay. So your kidney is metanephric, okay. Yes, clear. Yeah, actually you have this entire thing in MBBS where you will study everything, okay. There is cap formation, there is blastema. So all this will form later, slowly it will continue the development to form your glomerulus, loop of Henle, PCT and all that. But just as of now, just understand that there is pro, meso and meta. Pro will degenerate because it is at a very primitive stage. Then meso will start forming. Again, that also will not be completely functional. From the meso stage, meta stage will continue the development and finally form your this complex kidney, okay. So two terms I made you very clear, retroperitoneal and metanephric. Both the terms you will study in detail in your MBBS first year or second year, okay. Under embryology you will study. Okay, now it is just a trailer, like Laharika told, just a trailer, okay. Yes, okay. Students, we will just know the small things, where is it present and all that. So I told you about retroperitoneal space and peritoneum, no. So how does this happen is, basically, so students, imagine this is how the space is, okay. This is how your retroperitoneal space looks like. In that, what is going to happen is, your kidney, okay, your kidney will be here. One kidney, second kidney. This kidney will be attached in the, so here this will be the ventral side. Okay, this is a cavity, okay. I already told you about the cavity, so you should just assume that this is one cavity in my body. Okay, kidneys are attached here. Now we are talking about the covering. I told you peritoneum, right? Peritoneum will be there in the center, right? So that will do the covering of the organs. So now what is going to happen, students? So say kidney is attached to the dorsal side. Kidney is attached to the dorsal side. Now here, if you see, this is how your peritoneum is going to cover. So it will basically cover the kidney, okay, it will basically cover the kidney only in the ventral side, that's all. That's all to remember, okay. So your kidney will be only covered in the dorsal ventral side and not in the dorsal side because it is attached. If something is attached, you will put paint over here or you will go down and put the paint. You won't do that, no, you will just put like this, you will cover the top portion which is visible to you, that's all. So this is the reason why they say it is surrounded by peritoneum in the ventral side, okay. So students, the problem with your NCERT is terms will not be mentioned, but these terms you have to study. So that's the reason I'm explaining so much. It is surrounded, okay, surrounded by what? Peritoneum. Peritoneum, yeah, peritoneum. Which side? See, kidney is attached to the dorsal side of the cavity, but peritoneum is covering the ventral side of the kidney, okay? No confusion in that. Peritoneum on the ventral side. Ventral side of the kidney. Done? Okay. Next one, students, tell me the weight, width, length, all that. Can you tell me? Okay, come on. Yes, students, tell me the width, height, weight, all this you have to tell me. Come on. Important part. You open your NCERT and tell me. Okay. Yes, by then I'll write the rest. It is It is metanephric, retroperitoneal, yes. Tell me quickly, what is the width, height? Yes, good job. Okay, thank you, Jai Ganesh. Okay, students, yes, it is situated. Okay, it is situated. That means location is where? In the 12th thoracic to the 3rd lumbar. Okay, 3rd lumbar. Now, you people know the bones of all this. Okay, so you should always revise. Students, if you do not revise biology, there is no way you will remember, and that's the reason why biology is difficult. See, even if I've taught this chapter 2 months ago, you will not remember half of these things. Why? Because biology is a subject that you have to go back and read it every single time. Every time I take a class, I always go back to my notes, because even if I'm a teacher, even if I'm teaching this in, many platforms also, I cannot remember it. Okay, because that is how biology is. That is the reason why biology becomes difficult. Why not everyone is able to score 360. Okay, yes. Situated, location done. What will be the length and everything? Yes, very good. Thickness will be how much? 2 to 3 centimeter. Okay, and then width will be how much? 5 to 7. Yes, 5 to 7 width will be? Yes, very good. 5 to 7 centimeter. Length will be how much? Length will be how much? 10 to 12. Yes, very good. 10 to 12 centimeter. Okay, done. So, this is the general thing which is mentioned in your textbook. So, location 12th thoracic to 3rd lumbar, thickness 2 to 3, width 5 to 7, length 10 to 12. Okay, this is general thing about your kidney, and this is your peritoneum, your space. I know that only a student who is very keen to crack NEET will be able to sit throughout this class. Otherwise, they will not be able to. I know this for sure. Why? Because the chapter, sometimes at one point it is very dry. Sometimes it might be really good. But, there are ups and downs in this chapter, and only a student who is very keen that no matter what, however long it takes, I am going to sit for this class. Only those students will be able to sit throughout this class. Okay. Yes. Students, now we will be doing the longitudinal session of kidney. After that, I'll give you a kutti break. Okay. So, come on, let's do this kidney. All of you take all the color pens in your home and let's start the class. Okay. Yes. Yes, done. I wrote the weight of the, weight is 120 to 170, no? 170 grams. Yeah, this Sharath is like one CBI only. If he's in the class, I have to be worried. Like, ma'am, you didn't write this. Mr. CBI. Yes, 120 to 170. I wrote, sir. Are you happy? Okay, fine. Students, let's draw the kidney. Let's draw the kidney, okay? Very beautiful kidney. Let's draw. Come on, all of you. Draw with me. Students, I have literally not drawn anything, so I can draw it with you people. So, all of you also find it easy. Yes, this is not, very bad kidney. Okay, let's draw a little. Yes, this is not your elephant trunk, this is the kidney. Okay, this is the kidney. Now, ready? Done? Kidney done? Yes. Now, students, all of you know that kidney has two portions, cortex and medulla. Yes or no? Kidney has cortex and medulla. So let's draw one separation between them so we will be able to understand. Okay, very kutti separation let's draw. So students, two regions are there in the kidney. What are they? So kidney has two regions, cortex and medulla. Now we have to understand what is present in each of them. Okay, what is present in each of them? So we will start with your cortex. This is your cortex. I am differentiating with some dots I am putting here. Yes, okay, dots, fine. Now students, so first thing we will be drawing, okay, first thing we will be drawing is your medullary pyramids. Now students, understand, some portion of the medullary pyramids is in the cortex, most of the portion is in the medulla. Okay, clear? Most of the portion is in the medulla. Okay. Yes. Yes, medulla. This Anish, medulla is there everywhere, right? You study in ovary, you study in your adrenal gland, right? So medulla cortex, you will keep listening. That basically means two regions. That's all. Okay. So let's draw some pyramid structures. All of you draw with me. So one. There's no number as of now. We were just drawing for our understanding. Okay. Done. So we drew the medullary pyramids. Okay. Students, now this medullary pyramids, yes, okay, these are the medullary pyramids. Now I will draw something called as your papillae. Okay, here. Okay. These in between are your papillae. We will label it, do not worry, we will label it, but just as of now, understand so much. Okay. Students, on this medullary pyramids, you will have your nephrons. Okay. So, I cannot zoom and show, but I hope it is visible to all of you. Can you see nephrons on the medullary pyramids? Yes or no? I have drawn nephrons. See here, you can see the Bowman's capsule and then your PCT, DCT. Okay. Yes, fine. Now, we will start labeling whatever we understood. So, students, this region is your cortex. This is your cortex, okay. Then, in between, one line you see, no, in between the cortex and the medulla, there will be one portion which is called the column of Bertini. You can say renal column of Bertini or you can say column of Bertini. Done? Renal column of Bertini. Now students, other than that, what did we draw? We drew the medullary pyramids. Okay, let's label that also. This is the medullary pyramids. Now on the medullary pyramids, whom do you see? You see the basic structural and functional unit of kidney, that is your nephrons. Okay, so let's label that also. So you see your nephrons on the medullary pyramids. So if you see medullary pyramids, it is little going to the cortex, like the little portion is to the cortex, but the major portion is to the medulla. Okay. Then I drew some green color structures, right? Those are renal papillae. Those are renal papillae. Okay, renal papillae. Now students, understand this renal papillae will open to a very wider region. Can you see this wider region? This is actually your ureter, right? That wider region is called your renal pelvis. What is it called? Renal pelvis. Done? Not the pelvis there, okay? Renal pelvis here. Yes? Okay, fine. Now students, this renal papillae is this thick portion which I have drawn. This, can you see here the circles? This is your renal papillae which I have drawn. More you can draw if you have more space. Now, renal papillae will open into the minor calyx. Minor calyx will open into the major calyx. Major calyx will go to the renal pelvis. Okay, so let's write that. Let's write it as like a flow chart. So, students, renal pelvis is here. Now, it is like How you know? Like you have gullies, no? What do you call for gullies, yeah, in your languages? I don't know what you tell it in Malayalam also. Students, what do you tell for gullies? Students, tell me. Okay. Yes. Students, what do you tell for gullies? Yeah, gullies, I think Hindi students would have understood. Small streets are there, no? Small streets like small route map. I'm not able to get the word also. Small areas will be there, no? We call it gullies, no? So that kind of a thing is called your minor calyx and your major calyx. That means smaller ones are your minor one. The bigger ones are your major calyx. So what is happening from the renal papillae? So this is your renal papillae. Okay, this is your renal papillae. Renal papillae will open to your minor calyx. Okay, this small, like you know, like one gully you see, that is your minor calyx. If you know it in your language, please put it in the chat box. Okay, minor calyx. Then minor calyx will open here, right here, which is your major calyx. Okay, and this major calyx will open into the renal pelvis. Okay, renal pelvis. Colony, I don't know. Yes, allele. Okay, your streets or okay, so small streets, that's it. Okay, those are called as your gullies, right? I don't know why I'm not able to think of an English word for it. So let's write it. So what did I say? Let's write it here. Renal papillae, very, very important, students. Renal papillae. Okay, from the renal papillae, it will open to the minor calyx. Minor calyx, it will open to the major calyx. Major calyx, say it will open to the renal pelvis. From the renal pelvis, it will go to the ureter. Yes or no? Okay, whom are we talking about? We are talking about the urine, right? It will go from the renal papillae to the minor calyx to major calyx, major calyx to renal pelvis to your ureter, ureter to your urinary bladder. Is this clear? Easy? Okay. Clear, students? This much is clear? Okay. Now, yes? Yeah, colony is like this, no? But this one is street. Okay, no problem. We understood. So, if you understood, it's better. Okay, now students, this is your kidney. General we understood. Now, we know that there is a portion in the kidney where your veins will come. Okay, let's draw the veins. Then you will understand. Yes. Now, look here. This portion, your renal artery will come. Okay, renal artery will come. Same way, renal vein will come. Now, how do I draw the renal vein? Is it visible? No problem, no? Okay, let's draw it in single line. Okay, let's draw it in single line. So, it will be, I don't want it to be so clumsy that you don't understand. Fine. Okay, renal vein. So, students, there's a small point. All of you tell me in the chat box which is that place where this artery, vein, all this will open. Even some blood vessels will open. This is called your hilum. What is it called? Hilum, right? So, this spot where your, so hilum is a notch. Hilum is a notch where your blood vessels, right? Your notch where your blood vessels will come, as well as who is coming? Your renal artery, renal vein is coming, right? And even some of the nerves will pass. Okay? Some of the nerves will pass. Okay? Yes or no? Yes. Okay. So students, here hilum is the notch where your, okay, hilum is the notch where your renal artery will pass. Very important, okay, this question will come. Renal artery plus renal vein plus nerves passes. Okay, plus the nerves will pass. This is a spot where all of this will pass. Okay, now students, we have a covering. Now this is such a big kidney, we need protection, right? We need protection. So for protection, we have 3 layers. For protection, we have who? We have 3 layers. First one is called your renal capsule. Okay, renal capsule. Then you have adipose tissue. Then you have adipose tissue. Okay, wait. Okay. You have adipose tissue. Basically, adipose tissue will help with all the shock, right? Any jerk movement has happened, that will be done. Your adipose tissue will take protection of that. On the top of this adipose tissue, there is another spot called as renal fasciae. Okay, renal fasciae. Together, they will form the envelope. Okay, together they will form the envelope. So, can we label it? Can we label it? Yes, the first layer, okay, first layer is called your renal, what is it called? Renal capsule, okay. Second layer is called adipose tissue. Adipose tissue. On the top, whom do we have? Renal fasciae. Now students, together is called the renal envelope. What is it together called? Renal envelope. That means the protection. Okay, all 3 together. Okay, renal envelope. Is this clear? Is this clear? We drew a beautiful kidney. Yes, correct. Renal adipose, renal fasciae. Okay, very good. Okay. Done. This much is clear. So, this will be there entire all throughout the kidney. Okay, we are not drawing it because it will become too much to label and too much to understand. That's the reason we are not drawing everything. But students, let's do a quick recap. What if we have missed something? Nothing is missed, but in case. So, students, let's do a quick recap. We are taking a LS of a kidney. Okay, LS of a kidney we are taking. 1 second. Okay. So, students, we have taken a LS of a kidney. So, when you take the LS of the kidney, there are 2 regions in the kidney, cortex and medulla. There are 2 regions, cortex and medulla. When you see the cortex and medulla, you should understand that the cortex part, okay, in the cortex part and in the medulla part, there are some pyramidal shaped structures which are called as your medullary pyramids. On the medullary pyramids, you will be able to see your nephrons, okay, very important. Now, this medullary pyramids, after that, in between, like to the portion where the ureter is opening, right, that portion, we will have something called as your renal papillae. That renal papillae will open into the minor calyx, it will open to the major calyx. From the major calyx, it will open to the renal pelvis, and from the renal pelvis, it will open into the ureter. That means after urine formation, this is the pathway that they take, okay. Now, there is a spot which is, or a notch which is called as hilum, through which the blood, that is your renal artery, renal vein will pass. At the same time, even some of the nerves will pass through this hilum, this notch, okay. Then there is a mid line which is called the column of Bertini, okay, mid place which is called the column of Bertini, and this wide mouth like opening to the ureter is called the renal pelvis. Now the entire kidney is protected by 3 layers called the renal capsule, adipose tissue and renal fasciae. Together forms the envelope of the kidney. Okay, is this clear? Okay, we have covered the entire kidney. If there is any doubt, please feel free to ask me. Profanity, my God, full English. Profanity, oh my God. Thank you. Okay, yes. Fine. Thank you, Sharath. Oh ho. Rishabh, if you use this much colors only, you will understand. When kidney uses makeup, it seems. If I draw everything in white, you will be crying now. Telling, ma'am, I don't understand anything. Why have we drawn everything in single color? Yes, done? Okay, fine. Next slide. Students, will you label? Shall I give you some place? I'll ask you some questions. Ready? Okay, we'll label. Okay, tell me which is this spot. Part 1. Visible, no? Yeah, it is visible. Okay, then tell me which is this region. Okay, this region. Yeah, this region. And okay, this region done. Okay, tell me, can you see? Yeah, they can all see this. Students, tell me which is this region. What do you see this? Okay, 1, 2, 3. 1, 2, 3, type it together. Let's see. Okay. Very good, very good. They have already typed the answer, yeah? Very good. 2 is hilum. Okay, 1 is, okay, 2 hilum. Okay, fine. 3, okay, nephron. Fine. Okay, good job, good job, good job. So, 1 is what, students? 1 is medullary pyramids. One is medullary pyramids, very good. 2 is hilum. How did you tell it is hilum? Because you can see the nerves entering from there, right? So, hilum, not the nerves, your artery and vein. Okay, 3 is what? Nephrons. 3 is nephrons. Good job. Good job. Okay, all of you, good job. Yes, good job, good job. Now, students, is this much is clear, no? So, like this diagrams can come, okay, in your CBSE also, I mean, now 12th portion, forget. But then in case in your exam it comes, you should be able to label. Done? Shall we do ureter? Or we'll take a break and do? Come on, tell me all of you. Shall we take a 10 minutes break and do, or we'll finish off ureter and urinary bladder? Very small topics. Okay, are you all tired? We'll do this. After that, we'll take a break. Hi, Banot. So many days I have not seen you in my class. Where were you? Crazy dude, I did not see from so many days. Madhumati, I didn't see. Rupa, so many days over. Crazy dude, see. So many of you have joined. Where were you, all of you? I didn't see you in locomotion chapter. Where were all of you? All went missing. All my missing students have come back. Yes, okay, fine. Continue. Okay, done. Students, look here. So, here we are going to understand some muscles, okay, some muscles. When I teach you animal tissue, I will teach you in depth about this, but as of now, just understand there are some muscles, okay, there are some muscles. So, ureter, you know, it's a long tube and this tube will be carrying the urine, correct? They will be doing peristaltic movement. What will be they doing? Peristaltic movement. That means that movement with which the urine will be expelled out. So, ureter has some muscles, okay. So, the first one is called your transitional epithelium. That means the epithelium which will be able to do that movement, okay. So, that movement is given by your transitional epithelium. Who is it? First one is your transitional epithelium, who is going to help for the movement. That means for the peristalsis movement to happen, no, this person will be helping, okay. So, this transitional epithelium will be helping in the ureter stretching, the stretch, okay. That is because of the transitional epithelium, okay. Then we have Next layer is a muscular layer. What is the next layer? A muscular layer. A muscular layer which will actually be made up of longitudinal muscles. Okay, it's a muscular layer which is made up of a longitudinal muscle. Okay, students, little more in depth you will study in your animal tissues, but as of now just understand that there are 3 layers. Okay, 3 covering coatings on your ureter and your urinary bladder. First one is your transitional epithelium, which is basically stretchable. Second one is your muscular epithelium or your muscular epithelium, which is made up of your longitudinal cells. Okay, and the last one is your connective epithelium. Okay, last one is your connective epithelium. That will be on the muscular itself. This is your connective cells. Okay, connective cells or connective tissues, which is actually helping all these 3 people will actually help them for this peristaltic movement. Is this clear? Simple, very easy, students. Okay. Yes, transitional. Okay, transitional and your. Okay, clear? Yes, fine. So these 3 muscles, I will draw a little thicker so it is visible. It's not visible at all now. Yes, students, draw by the time I do editing. Okay, that is one layer. Second layer is your muscular layer. Yes, and your third layer is your connective tissue layer. Yes, is this visible now? I drew it too thin. Now it should be visible. Yes, done. Okay, connective layer. This is all about your ureter. It is basically helping it to pass the urine and also be a little about the stretching. Okay, little helpful in stretching. Now you have to understand the urinary bladder. Again, students, same set of muscles, but here you will study little more, some more concepts. Okay, so let's draw the urinary bladder. Okay, this is how your urinary bladder will look. Again, this also has the same set of muscles, students. First, it has a transitional epithelium. Okay, this one, your transitional epithelium. I'll draw for one side. Okay, transitional epithelium. Second one is your muscular. Now, students, this muscular layer which has is your detrusor muscle. So, it is important to remember. What is it? Detrusor muscle. We will label it. Yes. So, detrusor muscle is the muscle that is actually helping. So, students, what is this muscle actually going to do? They are going to do the job of contraction, right? Only if contraction happens will be able to do micturition. Right, we'll be able to do micturition only if contraction happens. If contraction does not happen, we'll not be able to do micturition. Agreed, all of you? So, this detrusor muscle will actually help in the contraction, so the urine is expelled out. Okay, so it is very important to remember the muscle's name. Now, the last one is your connective layer, connective tissue layer. Again, it is there like that only. Okay. So, here we have the connective tissue. And this detrusor muscle is the muscular layer because of which your contraction happens, right? Contraction has to happen from both the sides. This pushing has to happen for the urine to be expelled out. Okay. Now, to this what will come, students? Ureter will come, right? So, ureter will come one side, another side. Okay. So, here I'll show you something. Okay. And students, here, wait one second. Ureter. Since to this, okay, to this, there will be also the urethral opening, right? All of you remember this, no? There will be urethral opening. Now students, this will form a trigonal structure. What will it form? A trigone of bladder. That means 3 openings together. So we have one opening of the 2 openings of the ureter and 1 opening of the urethra. This will form like a triangular structure. Can you see a triangular structure? This triangular structure is called the trigone of bladder. What is it called? The trigone of bladder. That means 2 openings of the ureter plus 1 opening of the urethra. Together forms the trigone. Clear? Easy? Wait, let me see. Yes, very good. Trigone of bladder. Okay, very good. Now students, 2 ureter, 1 urethra. No doubt in this. Now students, this urinary bladder, right? This urinary bladder, let me just extend this. If I extend this, there are 2 sphincter muscles. Okay, 2 sphincter muscles. Sphincter muscle 1 and sphincter muscle 2. Okay, so we can label it as S1 and S2. Let's label it as S1 and S2. And this S1 is the internal sphincter, which is not in our control, involuntary. It is not in our control. So, internal sphincter, and second one is your external sphincter. So, external sphincter is in our control. Now, example, you are going and asking your teacher, or in your offline center, you are asking, ma'am or sir, can I use the washroom? So, that means your internal sphincter is not in your control. So, by then your urinary bladder would have maybe filled up around 500. Actually, the capacity is around 700 to 800 ml. Okay, imagine your urine has filled up around 500 ml and you have asked your teacher. By then your internal sphincter muscle will be opening, okay, almost opening. The teacher will tell, no, you cannot go. Go back and sit in your place. Now, what will you do? You will be like, okay, I will wait because it's only 500. It can go up to 700. So, 550, it will become 600, it will become 650 when it becomes this sphincter muscle would have opened. Now, the external sphincter muscle also will be telling, because your receptor, what is that? Your stretch receptor will send signal to the brain telling that urinary bladder is full. It is time for micturition to happen. Now, you will be like, ma'am, I need to go. If I don't go now, right? That's all. So, that is when your urinary bladder gets completely full and both of your muscles are not in your control at this point. Yes or no? Have you ever felt this? There will be a point where you cannot control. Okay? That is again now the external sphincter muscles will open. Clear? Easy? Sphincter muscle. So students, the question can come, which muscle, which among the sphincter muscle is in our control? You will tell external sphincter muscle is in our control, internal sphincter muscle is not in our control. Okay? Yes. So who will send the signal? Stretch receptor. Here there will be a stretch receptor which will send a signal to the brain telling that urinary bladder is full. Now you can relax. Right. So this sphincter muscles, if they contract, urine will not be expelled out. But if they relax, it will be expelled out. So you have to remember that urinary bladder has to contract, but your sphincter muscles have to relax for the micturition to happen. Okay, for the micturition to happen. So, I am writing it here. Micturition will happen when your sphincter muscles relax and the entire urinary bladder contracts, or your detrusor muscle contracts. Is this clear, students? Please do not write sphincter muscles and this one will contract. Wrong. Okay, they shouldn't contract like that. They will. Okay, clear? Clear? Fine. Yes, very good, very good. So, students, sphincter muscle has to relax, urinary bladder has to contract, or your detrusor muscle has to contract, then only the urine will be expelled out. So, do not write sphincter muscles has to contract. If sphincter muscles contract, urine will not go out. They have to relax, then urine will go out. Clear? Now students, there's a condition when your sphincter muscles, like in the sense when your stretch receptors does not send signal to the brain. That means stretch receptors have stopped working. Actually, when PYQ was asked on this, what happens if the stretch receptor stops working? Okay, that time the answer should be that once the stretch receptor stops working or is completely at fault, then your brain will not get a signal that the urinary bladder is full. How much ever it is getting full, you will not know. That will lead to, okay, that will lead to incontinence. Okay, incontinence. Okay, that means this will be there especially for people who are, you know, old age. Basically comes for people who are old age. What happens is their stretch receptor will start getting weak, like the signals will not be sent. That is the reason sometimes by mistake they will, you know, urinate at places and they'll feel very bad because they wouldn't have got to know that the urinary bladder got full. Is this clear? So incontinence is the condition where the stretch receptors fails and the urine will start leaking. Okay, urine will start leaking. Yes, clear? Yes, no signal. Exactly. No signal will be sent. So, the brain will not understand when it is full. So, because of that, what will happen? The urine will leak. Okay, this is mainly in people who have old age. Urine leaks. Okay, done. Now, we can take a break because after that, no, 1 second, I'll finish urethra also. Students, urethra, nothing is there. Just about the female and male. So, students, understand in urethra, if you see, urethra as a whole, if you see, males and females are a little different, okay? Female and male. Just about the structure I'm talking about, So, first understand in females, your urethra is actually short. Okay, urethra is actually short and in males it is long. And also understand in males it acts like a urinogenital, okay, urogenital duct or tract. Tract is the best word. Okay, so it is a urogenital tract. Okay, I think duct is mentioned in your textbook, so we'll write duct. Okay, so students, it is a urine. Why am I telling urinogenital? Okay, so it is urinogenital duct. That means both your urine and sperm comes out through the urethra. Okay, both of it, that is your sperm and urine will come out of the urethra and it is very long. In females, it is not. It is completely only carries urine. Okay, only for urine. Because females, the urethra is short. Females have high chances of UTI. Compared to males. Males also get, it's not like males won't get. Males also get UTI, but the thing is females have high chances of getting UTI because in females the urethra is short. Is this clear? Very small information. Okay, just long and short, just for you to understand why UTIs are common in females compared to males. That's all. Okay. Yes, very good. Okay. Is this clear? Any doubts? Any doubts? Okay. Okay, yes, urogenital duct, okay. Tract also you can write, no problem, where urine and sperm will be together coming out. That's all about urethra. Next we have about nephrons. So before we start nephrons, we'll take a break. Okay, we'll take a break. Students, how long you want a break? Will you have lunch and come? Or you want just a short break? Can you all tell me in the chat box? And those who join new, quickly hit the like button. Come on. Students, come on. Yeah, 100 likes also we can't make. So sad. So sad life. Yes. Students, some things are not there in NCERT. Very small things like how I told you retroperitoneal space, peritoneum, all that is just mentioned here and there, but explanation is not given. I am giving from PYQ point of view. If I have seen a PYQ from that portion, like how I told you crayfish, crawfish, I saw in PYQs. So, whatever I see in PYQs, I have added extra. Okay, other than that, everything is there in NCERT. Yes, tell me all of you. Short break? Kit Kat break, it seems. Short break. Okay, fine. Now, what's the time? Okay, 12:50. We'll be back by 1:15. Okay. Students, maximum 1.5 because I have to go all the way down. That's the reason. Okay, 1.5 works. Okay, all of you should be back. Don't be like, ma'am, oh, major part is not over. Now picture is left. We have a lot of mechanisms and all, right? So 1.5, all of you be back. Yes. Fine. Students, am I audible? Yes, hello. 1 second. Yes. And if you're back, the mic. Are you all back? Yes. Okay. Hello, hello. Not sure. Okay, let's continue. Students, share the link and tell them that the class is starting. Wait, I will just drop a message. Otherwise, they will. Yes, I put a message. Okay, students, so we are going to do nephron. Right? Okay. Okay. So we understood urethra, urinary bladder, all that we understood. Now our task is to understand nephron, right? Nephron is actually the basic structural and functional unit of your kidney. Yes or no? Yes. Basic structural and functional unit. Like how we have cell, right? We have everything. We have studied so many structural and functional units. Same way here, nephrons are the structural and functional unit of kidney. So they constitute the kidney. Now students, understand nephron basically has two parts. Nephron basically has two parts. Okay. First part is your glomerulus. Okay. First part is, 1 second. Yeah. Okay. So the first part is your glomerulus. That means which has your Bowman's capsule. Okay. Glomerulus, which is inside the Bowman's capsule. So it's a cup. Inside that, lot of capillaries are there, right? So it will look like this, right? This will be the Bowman's capsule. This will be the capillary. So it will look. So your glomerulus and the Bowman's capsule and all the renal tubules. All the renal tubules. These people constitute the nephron. Okay. Now renal tubules means who and all will be there? PCT, right? DCT and your loop of Henle. Okay. Loop of Henle. These three people constitute your renal tubule. And then your glomerulus will be placed inside the Bowman's capsule. Yes, all of you are back. Okay, great. So this much is clear, students. Then next one is what we are going to draw the entire nephron. Okay, we are going to draw the entire nephron. I want all of you also to draw with me. Okay, let's just draw the structure for our understanding. Now first we will start with your Bowman's capsule. This is how your Bowman's capsule will be. Extend it. 1 second. Draw it with a pen. Yes. So students, this is the entire tubule. It will be very, very convoluted. Okay. We cannot draw that much because then it will be too clumsy. Otherwise, it's very, very convoluted. Okay. So you can draw it however you want, no problem. Right? So I'm just drawing just to get an understanding of it. So you can also draw like this. But one thing I'm keeping in mind, I'm making sure that my DCT is very close to this. Okay. So I drew loop of Henle. I'm drawing my DCT and then I will draw straight structure. Yes, fine. This is your collecting duct. Okay, now let's draw the blood also, because blood has to come in, no? Otherwise, how purification will happen? If purification has to happen, blood has to come in. Yes or no? Yes or no? Come on, all of you smash the like button who joined right now. Okay, good. Students, so now we will draw the blood. Now, blood is coming from where? Blood is coming from your glomerulus, right? Capillaries. Capillaries are bringing the blood. So, let's draw your glomerulus. Okay. Now, look here. From the efferent, I am just connecting this. I think it has to be thin. Yes, 1 second. Yes. So, from here, we are connecting this. So, students, blood also can go anyways, okay. So, it is up to you how you want to draw it. So, I am drawing it this way. You can draw it anyways. So, now I am just connecting it, and from there, we will just take the connections back. We are trying to draw 3D, 4D and all, which is not possible. Okay. So, what is happening is here, when you come here, you make sure that you draw a U-shaped structure. You draw a U-shaped structure and you connect it anywhere, no problem. You connect it like this, you connect it like this, take it like this, no problem. Okay, just draw it somehow, just draw it. Okay. Yes. Now, students, we have connected the blood vessel in a random pattern. No pattern is there for this. Everyone can draw the way they want, but just we have to understand the concept. Okay. Now, let's label whatever we have seen. Okay. Are you ready? Yes. We will start labeling. Since my tongue is already twisty. Yes. Let's start labeling. Okay. Yes. So, what is this? First, whom do we have? We have the glomerulus. Yes or no? So, students, first labeling is glomerulus. Okay, then glomerulus is placed where? In a cup-shaped structure called as the Bowman's capsule. Right? Bowman's capsule. Then the first tube that you see, is what? PCT. First tube is PCT. Okay. Then the U-shaped structure, students, which you see is your loop of Henle. Loop of Henle. LOH I am writing. And now very interesting part, students, this blood vessels, no, with U-shaped structure you see, they will form something like this in the middle, which is called as vasa recta. They are basically capillaries, okay, peritubular capillaries which will run and form a vasa recta. So, I am labeling that also here. This is your vasa recta. It will be on the loop of Henle. So, just above the vasa recta, that means towards your ascending limb, okay, towards your ascending limb, you will see peritubular capillaries, okay. Peritubular capillaries. Okay. So, students, we have ascending and descending limb. This also you know, no? So, this will become what? Descending limb. It's important to know ascending and descending limb when we study urine formation. This will become the ascending limb. Okay, done? Done? Now, from peritubular capillaries, where are we going? To your DCT. From DCT, collecting duct. Students, I have a question. Which among this part is not a part of the nephron? Which among this part is not a part of the nephron? Come on, all of you can tell me. Which of this is not a part of the nephron? Yes? Which among this is not a part of the nephron? By then, I'll draw something. So, all of you tell it in the chat box. Yes. Go for it. Tell me. Very good. Collecting duct is not a part of the nephron. Why it is not a part of the nephron, students? Because many neurons open into one collecting duct, right? Many neurons will open, many nephrons will open into one collecting duct. That is the reason collecting duct is not a part of the nephron. Now, students, look here. Looking at this is a medullary pyramid, or if you remember this is the medullary pyramid and this is the nephron that I have drawn. Now, you tell me which part of the nephron will be in the medulla, which part of the nephron will be in the cortex. Okay, I have not drawn a difference here. That is the reason I am explaining you here, because if I draw one line, I think it will look too clumsy. So, tell me which part of it is the part of the medulla, which part is the part of the cortex. Jaldi bolo. Who is falling into the cortex, who is falling into the medulla? Okay, so all of you know this is the cortex region, this is the medulla region. Okay, yes, now all of you can tell. Come on. Go for it. Why is it slow? Come on, tell fast. Isn't it easy? PCT, vasa recta, ascending lymph. Yeah. Yes, students? Yes, okay, very good, very good. So, the only thing that will be in the medulla will be your loop of Henle, right? Loop of Henle will be the one that is, if you see here, that is the one, not the full loop of Henle, at least the descending lymph and the starting of the ascending lymph will fall into the medulla, the rest everything is in the cortex, okay? So, Bowman's capsule, PCT, DCT will be in the cortex, only the loop of Henle will fall into the medulla, very good. Why I am telling you this is because there are types of nephron depending on if it is in the medulla or in the cortex, or how much is in the medulla, how much is in the cortex. Okay. Now, students, nephron is clear. With this understanding, you have to do urine formation. You have to understand urine formation with this. Okay. So, clear? Yes, fine. Okay. Now, students, let's try to understand types of nephron. Where did I mention it? Okay, wait. We will write it here only. We'll write it here. Okay. Students, type of nephrons. One second, I'll just write it one side. So, we will write types of nephron also here. Yes. Ready? Did you all draw the structure? Yes, done. Okay. Students, now I told you, depending on who, like how much is there in the medulla, how much is there, loop of Henle, how much is there in the medulla, there are two types of nephrons. Okay, nephrons, two types of nephrons. By mistakenly, there are chances that sometimes I tell neuron because I just finished neural control and coordination. So, do forgive me for that. Okay, I mean nephron. Okay. Yes. So, students, depending on how much like short or long loop of Henle. There are two types of nephrons. So the nephrons are divided into two types. It is mentioned in your textbook if you notice. One is called cortical nephron. Okay, cortical nephron. These are the nephrons that is working in normal condition. That means now we are all in the normal condition. I am drinking water, eating food, everything I am doing, even you are doing all of this. Cortical nephrons will be in the normal condition. Then we have juxtamedullary nephron. What do we have? Juxtamedullary nephron. Students, juxtamedullary nephron works when we are in stress condition. Imagine now I am fasting. Okay, I am not drinking water, I am not eating food. During that time, to retain whatever is there in my body and for me to not lose any more water, that time who will work? Juxtamedullary nephron will work. Okay, so now students, how does this look like? So imagine this is the layer. So in the medullary pyramid, I am just drawing the thing. Okay, so look here. So we have one. Okay, yes. Okay, can you see two nephrons here? The smaller one, the small kutti pink one I have drawn is the one where the loop of Henle is short. Like what is happening is very small portion of loop of Henle is going into the medulla, very short loop of Henle. Okay, and in second case if you see loop of Henle is very big. Okay, very big. Now other than that they have one more thing. Vasa recta. I told you in the previous slide, if you see students, you can see, yes, here, see, Vasa recta is extending all the way to the loop of Henle. Yes or no? Now, any doubts? No, right? Okay. Students, Vasa recta is extending all the way to loop of Henle. In this one, what happens is Vasa recta will not, in which one? Cortical nephron, Vasa recta will not extend to the loop of Henle. So, the blood vessels will be like this. Okay, blood vessels will be like this. I don't think you can see it because I have drawn so small. But just understand. You see? So, Vasa recta will extend completely. Okay. So, what is the first thing that you understood? That it has short loop of Henle. Okay, very short loop of Henle. And second thing you understood is no Vasa recta. No Vasa recta will extend. And this works during normal condition, not during stress condition. Okay, normal condition. Now, second part, juxtamedullary students, this one has long LOH, okay, and they have Vasa recta. Okay, Vasa recta is present and this works during stress condition. Okay, it works during stress condition. Now, number also is there. Okay, number also is there. The number is cortical nephron will be more because usually normal condition is there. Okay, so cortical nephron will be around 85%. 85% of our body has cortical nephrons. I mean 85% is cortical nephron. The rest 15% is juxtamedullary nephron. Got it all of you? Any doubts? I don't think any doubts will be there. Very simple concept. Yes. Okay, great. So students, this much is clear, no? So types of nephron also we understood. It is mentioned in your textbook when we are studying juxtamedullary filtrate, but I am explaining in a flow. Okay, I am explaining in a flow. Yes. Fine. Now this much also we understood. Now we are going to understand the flow of blood. Flow of blood means, students, if you understand this only, you will be able to understand. Okay, no doubts, right? Okay, you will be able to understand like how is blood entering, how is the filtration happening. For that, you have to know the flow of blood. Okay, so shall we understand that? So students, first thing, so flow of blood if I am talking about, first understand from the aorta, right? From the aorta, blood is coming. Am I stuck? No, yeah. So from the aorta, blood is coming where? To renal artery. To renal artery. So from renal artery, blood will go to the kidney, right? But you should know in which area, where it is going. Oh, students, 1 second. I think I forgot to label this. Students, this one, no? Oh, this mic. Yeah, this part, this portion which is coming in is called as efferent arterioles. Right now I'm writing AA, and this one is called efferent arteriole. So which will leave the kidney is efferent, and which is coming is called efferent. Okay, keep in mind. Yes. So students, renal artery, say blood will come to where? Blood will come to efferent. Efferent arteriole. From efferent arteriole, where will it go? It will definitely go to the glomerulus. Yes or no? Right? Glomerulus. Now from glomerulus, where will it go? It will be taken out by the efferent arterioles. Okay, efferent. Okay, efferent arterioles. Students, so this was clear? Efferent arterioles. So, aorta, renal artery, efferent arterioles, glomerulus, efferent arterioles. Okay. Now, students, from efferent arterioles, I showed you peritubular capillaries. Do you remember? So, it will go to the peritubular capillaries. You should think of the diagram that I drew. That time you will understand. From efferent it was coming down, right? So what was there? Peritubular capillaries was there. From the peritubular capillaries, it will go with the help of renal vein. It will be taken, right? So with the help of renal vein, it will be taken out. Okay. So here what is happening, students? This is I told you that this blood is oxygenated, but it has toxins. Toxic substances are there. What is the toxic substance? Urea is there. But this blood will be deoxygenated. That means other chemicals will be like oxygen, all this will be lack. Deoxygenated, but here no toxic substance will be there. That means basically no urea will be there. Is this clear to all of you? Okay, is this clear to all of you? Yes, very good, Subhashini and Sanvi. No, Banot, afferent come efferent, E. A comes, E goes. Okay, A comes, E goes. Yes, very good. Deoxygenated blood, no urea. Oxygenated blood, urea is there. Okay, this is the pathway of your blood. Okay, so the blood will enter and this is how it will go out. So students, if you look at this diagram, no? If you look at this diagram, here, somewhere here, okay, after the peritubular capillaries, you will be able to see renal vein. Okay, when renal vein will enter like this. Okay, is this clear? One renal vein will enter like this. Yes. Okay, done. Now, next one is juxtaglomerular apparatus. So, students understand, whenever there is a problem with the filtrate, problem with whom? Filtrate. Glomerular filtrate is coming, yes or no? So, whenever there is a problem with the filtrate, who will recognize it? There are specialized cells called macula densa. But just try to understand it very simple. Yes, very good. Now, try to understand it in a very simple way. Okay, very simple way. 1 second. Yes. Can you all see clearly now? There's a reflection coming, no? Okay. Students, so look, always we have to go back to this diagram. Okay, always go back to this diagram. I told you, I am keeping in mind one thing. What was one thing that I kept in mind? That my glomerulus and DCT comes very close to each other. I told my glomerulus, my Bowman's capsule and DCT has to come very close to each other. Why did I say this? Now you will understand. So students, imagine filtrate is coming in. Okay, this is a special kind of apparatus which works on understanding if the filtrate has kuch gadbad. Like in the sense, salt will be less, water will be more, water will be less, salt will be more. Any kind of things can happen in the filtrate. Now, who will recognize this? That the filtrate, glomerular filtrate is coming, it has some problem. For that to understand, there is a complete whole mechanism. So, what you're seeing here is your Bowman's capsule, right? It is inverted. So, we are drawing here Bowman's capsule. Okay, Bowman's capsule. Now, this one which you see. So students, you just invert it and keep it. Okay, if I draw inverted, the problem is I cannot draw DCT. It will be wrong, right? If I keep it like this, right? Generally how it is like this, no? Like this, if I am drawing, it will be wrong because DCT won't come here. DCT will come downwards, right? So that's why this is drawn ulta. Understood? The confusion? Do not have a confusion why ma'am drew it opposite way. Now, this is Bowman's capsule. What will be this? This is the one which is coming in. So tell me what will be coming in, efferent or efferent? Efferent or efferent will be coming in? Efferent will be coming in. Yes or no? So students, this will be your efferent arterioles. This will be your efferent arterioles which is going out. Now, in the efferent arteriole, there will be some cells, some smooth muscle cells which will modify itself to form something called as the JG cells. What will it form? JG cells, juxtaglomerular cells. Okay, efferent arteriole, try to understand. Some of the cells of the efferent arteriole will modify themselves. Okay, is this clear? Yes. Okay, one second. Those who are attending this class first time, like first time you are studying excretion, is this clear to all of you? Okay, some of the cells of the efferent arteriole will modify themselves to form some of the smooth cells, okay, form JG cells, juxtaglomerular cells, okay. Now this juxtaglomerular cells, let them be there. Now this one, what is the circle I have drawn? This is a portion of your DCT, portion of your DCT. So in DCT also there will be some specialized cells called macula densa. What does it have? Macula densa, okay. So this is your DCT. This is macula densa. Basically, this entire system is to check on what is the filtrate that came in. Is the filtrate okay or not? Okay. So, DCT has specialized cells called macula densa. These macula densas are actually sensor cells. What will they sense, students? They will sense if the filtrate has some problem or not. Okay. They will sense, the macula densa will be able to sense if the filtrate has some gutbud or not. If the macula densa sense that there is some problem, what will it do? It will send the signal to the JG cells. It will send signal to the JG cells telling that some problem is there. Some problem is there. So, when they realize that some problem is there, this JG cells will release something called as renin. Okay, it will release something called as renin, which will kind of help in fixing the problem with the filtrate. Is this clear? All of you tell me in the chat box, is this clear? Those who are learning it for the first time. Is this clear, students? Clear? All of you? Okay. So, we will just write, okay, we will just write very little portion of it. So, we will just write efferent arterioles will, some of the smooth cells will get modified. I am not writing the whole thing, but at least for you to understand, it will form JG cells, okay. Now, if there is a problem with the filtrate, that will be recognized or identified by whom? By the macula densa. Macula densa, which is in the DCT. This macula densa will send a signal to whom? It will send a signal to JG cells telling that there is a problem with the filtrate. So, you do something. So, JG cells, what it will do? It will ultimately release an enzyme called as renin. Okay, it will release an enzyme called as renin, which will help in RAS mechanism. What will it help, students? That mechanism you will study next. Do not worry. It will help in RAS mechanism, which will actually fix half of the problem of the filtrate. Is this clear? Is this clear? Once again? Okay, quickly look here. So, this is basically your neuron. Okay, sorry, nephron. Nephron, what is happening? Your Bowman's capsule and the DCT is very close to each other. Okay. Now, students, sometimes or sometimes what happens, the filtrate which is coming in, that is your glomerular filtrate which is coming in for urine formation, might have salt more, water less, water more, salt more, anything can come, okay, blood, because of which blood pressure can be low, it can affect a person, okay. So someone should be there to identify this mechanism, right? So for that, what has happened in the afferent arterioles have some smooth muscle cells. What does it have? Some smooth muscle cells. Those smooth muscle cells will get modified to form JG cells. Okay, this will modify to form JG cells. Now, this JG cells will be present in the afferent arterioles. Okay, no doubt in that. This is your glomerular. Now, in DCT, there will be some specialized cells called macula densa. This macula densa have the capability, they are sensor cells. They will identify that, oh, this filtrate has some problem. So, when they identify that the filtrate has a problem, what will they do? They will send signal to the JG cells telling that you please release the hormone called renin. Now, students, what renin is going to do, you will understand in RAS mechanism. As of now, understand how renin came. Is this much clear now? Okay? Yes, renin angiotensin mechanism. Yes, that you will learn. So much is clear now? Simple, basic. Okay, basic we have done. More you will study in detail. It's dark. Did you understand? Okay, students, don't faint seeing this. It is basically your how your JG cells, that your apparatus looks like. Okay. Now, if you see here, this is your macula densa. This is your macula densa. Okay. This macula densa. And if you see here, this is your glomerulus. Okay. Glomerular capillaries are there. Okay. Glomerular capillaries are there. This is your efferent arteriole and afferent arterioles. Okay. Now, here you have myocytes and all that. Don't need all this. Now, if you see here, students, you will be able to see some modified cells. Okay. Now, this is how it is. It is very complex structure. But now you will only study basic. You will only study basic. So, here you have macula densa. Here you have JG cells. They will send signal and they will release renin. Okay. So, here if you see, this is the whole portion of your glomerulus. This is how it will look. Now, please remember, parietal layer, podocyte, visceral layer, these all, basement membrane, these all you will study now in glomerular filtration. Glomerular filtration. Okay. Done? Done? Ready for a quiz? Ready for a quiz? Okay. Let's label. Okay. This, I want you to tell me this. 1. Okay. Okay. One second. I'll modify this diagram a little. Label this, okay, and label this. 1, 2, 3. 3 labelings. Yes? Okay, clear? Okay, fine. Yes, students, please look here. Label 1, 2, 3. If you have any doubt, please ask me. Label 1, 2, 3. Yes, 1 is efferent arterioles. Very good, very good. 1 is efferent arterioles. 2 is vasa recta. Very good. And 3 is DCT. Okay. Now, one more question to all of you. Tell me the modified cells in DCT. That is the question number 4. And tell me the modified cells in efferent arteriole and what it secretes. Question number 6. Question number 6. 4 is what is the specialized cells in DCT. 5 is what is the specialized cells in efferent arterioles, and 6 is what is the hormone that it gives out. Come on. Yes, correct. Very good. 2 is vasa recta. 3 is DCT. Okay, now students answer the 4th, 5th and 6th question. Yes, 4 is macula densa. Very good. 4 is macula densa. Macula densa, which are sensor cells. Okay, and then here 5th one is what? JG cells. Very good. Fifth one is JG cells. Sixth one is renin. Clear? We understood the entire structure. Okay. Now, how many marks did you get? Previous you had 3 questions. Now you had 6 questions. So, 6 plus 3, 9 marks. Okay. Whoever got 9 marks, put it in the chat box. Okay. 1 mark question, students. 4 mark questions I have put later. Okay. 1 mark question. Okay. By the time you put your marks, I will just draw the structure. So, it will be easy for us. Done, students? Did you write your marks? Did you write your marks? Oh, most of you got 9. Very good. Okay, no problem. Some of you got 6. Yes, very good. Most of you got 9. Okay, students, so now we will start urine formation, main topic of this chapter. So, urine formation basically happens in 3 steps. Okay, so let's write the step. First step is called as glomerular filtration. Second one is what, students? So, second one and third one both are tubular. Both are tubular. Okay, tubular reabsorption and tubular secretion. Okay, now let's try to understand this. So, students, 3 steps clear. Now, this is the glomerular. You can see this. Here you can see afferent and efferent is coming. Okay, afferent is coming, efferent is going out. Now, students, understand when you talk about glomerular filtration, what is happening? Basically, blood is coming in, right? Blood is coming in. Now, from the efferent, okay, blood is coming in and that blood is filtered. It is waiting for getting filtered. Yes or no? Now, this blood will go like this as filtrate. Yes or no? It will go as filtrate. So, that will become what? Glomerular filtrate. The blood which is going will become, this first step will be here. Okay. This will be called as your filtrate. Okay. GF. Not girlfriend, glomerular filtrate. Okay. Glomerular filtrate. Yes. Okay. Banot, we will try to finish the class as soon as possible, but I don't know how long will the class go. We have again some mechanisms and counter mechanisms left. Okay. Yes, it is pretty long class. I told you, long chapter, student, more than locomotion, this is long. Okay, so students, this is the first step. That means glomerular filtration, through the glomerulus, filtration will happen. Right. Second one is tubular reabsorption. Now, what will happen? Glomerular filtrate around 180 liters. Okay, how much? Around 180 liters of GF is formed. Glomerular filtrate is formed. Now, can 180 liters be urine? No, definitely not. That means this has so many useful substances that are required. All that has gone out. Now, for that to, to rectify that, we will do the second step called as tubular reabsorption. What will we do? Tubular reabsorption. So, look here, blood vessel. So, from the tubules, we will reabsorb it to the blood vessel. So, tubular reabsorption will happen. Let's write second step. Okay, let's write second step. Understood? Tubular reabsorption is happening. Now, students, sometimes the blood, what to tell? pH will be disturbed. Okay, pH will be disturbed. It could be that some ions are more in the blood, some ions are less in the blood. Anything could happen. So, in case ions are more and the pH is disturbed, okay, pH is disturbed, then what it will do? From the blood, whatever unnecessary ions are there which are disturbing the pH will go back into the tubule, which is the third step, tubular secretion. Is this clear? Is this clear? Okay. Yes, students. So much is clear. We understood the entire urine formation in 3 just small terminologies. Okay. So, glomerular filtration means the blood which is coming has to form as filtrate and it has to move through the tubules. First step, glomerular filtration. In that filtration, we will get a filtrate called the glomerular filtrate. Second step is what? From the tubule, in case 180 liters of glomerular filtrate is formed. Now, all this will not go as urine. Maximum 1 to 1.5 liters will form as urine. That means the rest, everything is useful substance. So, we will reabsorb that useful substances from the tubule to the blood. That is your second step. Now, the third step is what? Tubular secretion. That means sometimes the blood pH can be altered. That means some ions are more or less. That time, this blood will leave, right? That blood will send whatever is unnecessary to the tubules back. That is called as tubular secretion. Is this clear? Easy? Easy? Yes. Okay. Easy. Fine. That is your entire urine formation. Okay. Now, we have to study a little more in depth about your glomerular filtration. So, I will just write few things I wanted to draw, but it will take a lot of time. So, I will not draw that, but instead I will just draw that also very fast because I don't feel satisfied if I don't draw and teach. Since this is the fastest drawing I can draw, just to make you understand, okay? Okay, not bad. A glomerulus I drew. Okay, now, Bowman's capsule. Now I'll draw a glomerular. Okay. Okay, all of you look here. Now this is, students, so glomerular filtration. Now don't get it confused. Understand this portion, no? This glomerular, here, if you see, glomerulus is there, Bowman's capsule is there. Between this portion, the filtrate has to move, right? So there is a glomerular filtration setup, the entire setup which will be helping. I told you, when I showed you this diagram, I told you, please remember this people. Basement membrane, podocyte, visceral layer. Why? Because these layers are actually going to help for glomerular filtration. Okay, understood? Now that I have drawn in a very simple way. Okay, it might look like a tree and a reflection, water, sunset and all, but it is not all of this. It is actually your glomerular filtration. Okay? Okay, fine. Yes, good job. So students, let's try to understand which are this path. First, the outside part. The outside part of the Bowman's capsule is called your parietal layer. What is it called as? Your parietal layer. Students, if you are hungry, please tell me. I will take a break. I'll give you a break. Parietal layer. Now, the inside layer, right? Inside layer. Now, I have not drawn the inside layer. Why? Because the connection is not there. Wait, we'll just draw the connection. Now, the problem is, if I draw the connection, okay, students, let's just connect it like this as of now. Okay, let's just connect it like this. Yes, we made, there's always solution for all the problems. Okay, so it will actually connect it. Okay, so that is this portion, right? This portion, this inside portion is your visceral layer. Visceral layer. Students, this is only a diagram to make you understand. This is not how it is. Actually, it's a very complicated layer. I just showed you, right? So, yes. Now, this much is clear. Now, the blue color one which I have drawn is your basement membrane. Basement membrane. They have squamous epithelium, squamous cells. Okay, basement membrane. Clear? Now, what I have drawn here is your glomerulus. That also you would have understood by now. This is your glomerulus. So, in the glomerulus, there are spaces which is called as fenestra. Okay, what is this? These spaces are called as fenestra. Write it here. Fenestra. Fenestra. Okay. So, students, where and all it's going? From fenestra. Okay, this is clear. Basement membrane clear. Fenestra clear. Yes. Okay. So, here again afferent arteriole, efferent arteriole. Okay. Now these cells, no, which looks like a tree, they are called as interdigitating cells. That means like this, interdigitating. Okay, interdigitating cells. Those are again the gaps are called as podocytes. So there, these cells will be called as podocytes. So what is basically happening, do not worry, there is nothing big happening, but understand, blood will come like this through the fenestra, they will go to the basement membrane, through that it will go to the podocyte. So that is what they are trying to show. Okay, the thing what is that trying to show is that this is actually your squamous epithelium. Squamous epithelium. Students, this chapter has a lot of, theory type of a thing. What do you tell, like events, like how do you tell events, no? There are so many events are there. So, do not get yourself confused. Okay, do not get yourself confused. Okay, so yes, students, these are filtration slits. These are called as filtration slits and the spaces in between are called as podocytes. Okay, spaces in between are called as podocytes. So, how will it happen? Think very simple. Blood is entering through the fenestra. So, I am writing ulta. Okay, through the fenestra, it will go, 1 minute. Yes. So, students, blood is coming through the fenestra, it will go where? Basement membrane. From basement membrane, where will it go? To the podocytes. Okay. Now, in the this podocytes will later again help for the filtration slits. Understood? Understood? This is ulta. You should think like this blood is going, not like this. Right? Bowman's capsule is there, glomerulus is there. Okay, is this clear, students? Simple? Simple? Yes. Okay. Yes, gap between endothelium is fenestra. Sharath, what are you telling? Okay. Yes, students, this much layers you understand. Please understand, glomerular filtration is a non-selective process. Very important. It is a non-selective process. That means it will not filter out anything. It is non-selective. It will be like whatever comes goes. Right? Non-selective process. Clear? Okay. The only thing that stops from this layer are larger proteins. Larger proteins. Okay, like when you filter chai, no, in case there is a chai patti, that means a tea leaf, that will get stuck. The small things will pass on, no, same way. Non-selective process, all the smaller ones will go, only the larger proteins will get stuck. Clear? Yes, yes, many events and terms. Lot of events, lot of terminologies. But students, please try to understand this. This is a little complex topic for most of them, but I want to make sure that it is clear for you. So, this is the filtration membrane, okay. So, think of it like different layers, okay. So, students, this is one layer of your filtration. Imagine one layer is there, one layer you have made, in between you have kept squamous epithelium. Spaces are there, through that blood will go. That space is called fenestra. Through that, the blood is going through a basement membrane. That means a gap between the Bowman's capsule and the glomerulus. Through the basement membrane, now the blood will enter into the podocytes. From the podocytes, it will go into the filtration slit and directly it will enter into your PCT and it will go later to the tubules. Okay, that much you understand. That much you understand. Okay. Done? Yes, sir. Yes, in your textbook it is given epithelium of the Bowman's capsule and endothelium of your glomerulus, right? Yes, okay. This layers only. Yes, this is clear? This is clear? Yes. Now students, we have to understand one thing about pressure. Who? Not your exam pressure, not other pressure today. Okay, pakka, done. Okay. Fine. Now we have to understand, due to pressure difference, the filtrate which is coming in, going out, all that, thoda difference will be there, no? So, in glomerular filtration, students, only glomerular filtration is little, so many terminologies are there to remember, but if you come to tubular secretion and tubular reabsorption, only some ions you have to remember, okay? So, students, yes, so here what is happening is, so due to difference in pressure, the amount of filtrate that comes in and goes out has a calculation. So due to pressure difference, Two types of pressure is there. Everything has a positive and a negative. Yes or no? Every human also has a positive and negative. Same way, even this one has a positive and a negative. Okay. Now, there is a positive pressure and a negative pressure. Okay, positive pressure. Let's talk first about positive pressure. That means positive pressure is the one that favors the filtration. That will be like, okay, more filtrate come in, I will support you. Come in, they'll tell, no? Some people loves guest coming home. So they'll be like, come in. It's okay, I will make extra dal, I'll make extra sambar, I'll make extra rasam, they'll tell. There are some people who does not like guests. As soon as guests come, they'll be like, oh, when will they go? That kind of a people, no? So there is positive and negative. So this is the positive pressure. Okay? Yes, okay, good job. GHP. So the positive pressure is glomerular hydrostatic pressure. So sad of biology people, no? We have to study physics, chemistry also. See, hydrostatic pressure. Okay, so glomerular hydrostatic pressure. It's not fair on biology students that we have to study physics and chemistry. Yes. So students, glomerular hydrostatic pressure is the one who is actually helping in telling that come more blood come in, more whatever filtrate wants to come in, you come in, I do not have any kind of problem. Now, how is this happening? It's basically happening because of the differences. Okay, it is this glomerular hydrostatic pressure is mainly because of the difference in diameter of who? Difference in diameter of your arterioles, efferent and efferent arterioles. Okay, difference in diameter of efferent and efferent arterioles. Simple. That means if more filtrate has to come in, efferent arterioles diameter should be little more. That's all simple. Yes. Now students, this has calculation. So GHP is how much? How much can all of you tell me? Is it 60? Exactly. No, they have put physics and chemistry and biology also. That is my problem. Exam to okay, I understand. Okay, 60 or 55. Okay, mmHg. This one just clear, student. This is the positive person. He's like, he does not have any problem. How much of a filtrate wants to come in, come in. Now we have the villain in the story. That is your negative pressure. Villain is who but negative pressure. Now negative pressure also, they are double strong villain. Two people have come. First person is BCOP. I've taught you this when I told you about your body fluids and circulation. I told you BCOP. Okay. So student, this is blood colloidal osmotic pressure, full form. Blood colloidal osmotic pressure. Students, basically what is happening, colloids will be there. Yes or no? Okay, colloids will be there. This colloids will be like water, you cannot come in. It will start blocking the water. Who are the colloids? Like albumin. Okay, remember when I taught you in the plasma, I told you albumins are there, right? Fibrinogens are there, globulins are there. So in that albumin is there, no, which is there in the blood. It will be like, no, enough of water, don't come in here, please. So it will make that faces and it will not allow water to come in. That is called blood colloidal osmotic pressure. That means to maintain the osmoregularity, to maintain the osmolarity of the blood, this colloids will stop water from coming in. But this is a negative pressure. Clear? Clear? Yes, very good. Okay, blood colloidal osmotic pressure. This is mainly because of the colloids in the blood. Like example, I'll write here albumin. Main person who is doing this, stopping the water from coming is albumin. Okay, it will basically, stop H2. Yes? Okay, now BCOP also has a pressure value. BCOP is somewhere around 30. How much? 30 mmHg. This is how much pressure is exerting, negative pressure. Students, second one is your Second one is another person who is your capillary hydrostatic pressure. Now what are they doing? Students, understand there is already filtrate in this glomerulus. It's not like full filtrate is gone. Some filtrate is there. That filtrate will be like, already I am there now, why are you coming inside? Yes or no? Correct? Now elder sibling will always be like, why are you giving so much love to younger siblings, right? Same way. This already present capillary filtrate will be like, I'm already here now, why are you coming in? Don't come in, get out. So that is called as capsular hydrostatic pressure. Okay, what is it called as? Capsular hydrostatic pressure. Yes or no? So this is formed because of the already existing. Okay, this is because of the already existing filtrate. Okay, this is actually less compared to this one. Okay, their pressure is CHP is equal to 20 mm of Hg. Clear? Students, clear? Yes, okay, great. Now we have maths also. Out of all this, physics also we learned, chemistry also we learned. Now we have to learn maths also. Come on, let's do maths. Yes, all of you calculate and tell me how much will be the pressure then. By then I'll sit for some time. How much will be the pressure, students? Just calculate what will be the pressure. Just do minusing 60. No, maths is coming. We have to do maths. Yes, very good. Net filtration pressure is HGP minus BCOP. Where will we write, students? We'll take next page. Yes. So, what is the maths? So, we have to do positive and negative minus karna hai, okay? So, that will be what? Effective filtration. So, effective filtration pressure you can tell, net filtration pressure you can tell, no problem. You can tell net also. Okay, net filtration you can tell. So, how much it will be? It will be basically GHP minus BCOP plus CHP. So, both the negatives we are putting together and one positive say we are minusing it, okay? So, it is EFP is equal to how much, students? 60 minus 30 plus 20 is equal to 60 minus 50, that is 10. So, this is how much pressure is enough for the filtrate to enter, okay? This is enough for the glomerular filtration to happen. Only 10 mm Hg is enough for glomerular filtration to happen. Clear? Understood? Yes, very good. 10. Okay? Done. Okay. Students, so glomerular filtration you understood. Now, we will just see one diagram and try to learn what and all are getting excreted out. Then we will see NCERT ones. Okay. So, students, I told you if there is, okay, let's keep that aside. Okay. Now, look here. Here filtrate is entering, that is your from the glomerular filtrate PCT, it is going to the PCT. Here you can see what is happening actually. Here tubular, your reabsorption will start happening, okay. Whatever is needed for the blood will start taking out, because how much is formed? 180 liters are formed. Do this 180 liters have to go anywhere? No. 180 liters if it goes, then we are going to die, right? Only 1.5 to 1 liter urine will go out. That means all your H2O, glucose, amino acid, urea, all this will be reabsorbed. Okay, all this will be reabsorbed. Clear? Yes, got it? Okay, fine. So students, we'll just try to understand here. Here you can see a lot of ions are going out, right? HCO3, H2O, K plus, H plus, NH3. Here in descending loop, if you see, water is going up because this one is permeable to water. That means it will only remove water. It will not remove anything else. And if you look here, in the ascending lymph, it is permeable to NaCl. That means it will only reabsorb NaCl. It will not reabsorb water. So this is one mechanism which is made sure that the like quality is increased. If both of them are reabsorbing water and sodium level, then everything will become gut-bud. So that's why this, your descending loop will only take H2O, okay, only reabsorb H2O, and your ascending loop will only reabsorb sodium chloride. Okay, here also you can see same ascending loop, and here you can see in distal convoluted tubule. Now we will try to understand with NCERT. Okay, we'll try to understand with NCERT, but before that, one second. Tubular reabsorption, students, you have to know one more thing is about the threshold. Threshold. Remember threshold, all of you? Yes, okay. So students, tubular reabsorption, no, happens like in the sense there are some things which we need high threshold to absorb it back. There are some things which can be absorbed in low threshold. Some are there with no need threshold at all. That means no need that pressure, right? So tubular secretion, we have three things happening. Only this you understand, rest we will understand from NCERT. Okay, so we have high threshold. Okay, high threshold. Low threshold, no threshold. Okay. Okay, no threshold. Clear? So here what is happening is we have to see which are the things which will be absorbed back in high threshold. That means maximum reabsorption will take place of these substances. Okay, who it will be, students? So we have first glucose, water. Here you can see many of the ions, okay, but then you understand some important things I am writing, rest we will, rest no need as of now. See, Na, Cl minus and amino acids. These are the people that will be reabsorbed, maximum reabsorption will happen of glucose, water, sodium, chlorine and amino acids. Okay. Now, low threshold ones are the ones which will be reabsorbed. Okay. First one is urea, second one is uric acid. These two will be taking only low threshold. Now, the urea is for osmolarity. Okay, urea is for maintaining the osmolarity or for osmoregulation. Now, non-threshold ones are which one? The other, other waste materials, students. Other nitrogenous wastes are there, no, creatine and all that. Those things will be absorbed here with, it is not required much. So, very least reabsorption will happen. Okay. Yes or no? Is this clear? Yes, very good. Partial reabsorption, you have urea and uric acid. Yes. So, this is for you to understand easily. Okay. Yes. Then I told you about your loop of Henle. Loop of Henle, what did I tell you? The water is reabsorbed, right? The walls are not most permeable, right? That also you have to remember. Yes. So, students, your reabsorption 70 to 80% will be reabsorbed in your PCT. You know that from your NCERT, I believe, right? PCT, reabsorption, maximum reabsorption will happen. Okay. Yeah, what I was telling is about your descending loop. So students, descending lymph, no? I told you what is descending lymph. It is actually permeable to water. Okay, very important. It is permeable to water, but it is impermeable to the salts. It is impermeable to salts. Understood? Impermeable to salts. Okay. This is how they balance it. Okay. This is how they will balance it. So, the blood osmolarity should be always maintained up to 300. Blood osmolarity will be maintained up to 300. This I think is mentioned in your NCERT, no? Yes. Shall we do NCERT? Ready? Okay. Okay. So, look here students, let's try to understand. Such a big paragraph, we will only take what is important. So, the first step, okay, important. First step of urine formation is glomerular filtration. Okay, yes. So, on an average, 1100 to 1200 ml of blood is filtered by kidney. Okay, 1100 to 1200 ml of blood is filtered in the kidney per minute. Okay. Now, which constitute roughly around 1/5 of the blood. Okay, total blood that is pumped into our heart, right? So, out of that, this is around 1/5. Now, students, the glomerular capillary blood pressure causes filtration of the blood through 3 layers. What are the 3 layers? Endothelium of the glomerular blood vessels, the epithelium and the basement membrane, no? Epithelium and the basement membrane. Okay, okay, understood? 3 layers will constitute glomerular filtration. In that, we also studied fenestra, filtration slit, podocytes, visceral layer, parietal layer. We learned a lot of it because for NEET point of view, it is important. For CBSE, like exam point of view, 11th students, you can stick on to this. Epithelium of the Bowman's capsule, endothelium and your basement membrane. Now, the epithelial cells of the Bowman's capsule are called as podocytes. Okay, the epithelial cells, podocytes, you remember? Arranged in an intricate manner, that is interdigitating manner, okay, intricate manner, so as to leave some minute spaces called as the filtration slit or your slit pores. Okay, this is also mentioned in your NCERT. Okay, only that visceral and all that I don't think, yeah, it's not mentioned. Now, blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass into the lumen of the Bowman's capsule. Students, only the large proteins will not get passed. Large proteins will not get passed. This is called as ultrafiltration. Why is it called ultrafiltration? Because everything is getting filtered out so finely, so beautifully that we call it ultrafiltration. Samajh aa gaya? Understood? Easy? What is this number here? I told you, no, only real dedicated students will be able to stay for 3 hours, 4 hours. Others all are fancy. Yes, ma'am, I will do, ma'am, but won't do anything. Right? So, yes, students, clear? All this we discussed. Is there any point that we didn't discuss? We discussed everything. Okay? Now, next one, look here. The amount of filtrate formed by the kidney per minute is called glomerular filtration. That means the amount of filtrate that is formed in the kidney per minute will be called as glomerular filtration rate. Okay, GFR. GFR in a healthy individual is around 125 ml per minute. These numbers we didn't write, so please make a note. Okay, that's why I've put NCERT. That is 180 liters per day. 180 liters per day, out of which only 1 to 1.5 liters of urine will be excreted out. That means the rest full will be reabsorbed. Is this clear? Okay. Now, the kidneys have built-in mechanism for regulation of glomerular filtration rate. One such efficient mechanism is your JGA, glomerular filtration apparatus. Okay, that means students, just to regulate, just to maintain the mechanism, right? Renin will be secreted, you will understand in RAS mechanism, but all this is to regulate, right? So, JGA. JGA is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the efferent arterioles. Now, in their contact, a fall in GFR can activate the JG cells. A fall in GFR or some problem in your glomerular filtrate will activate the JG cells to release renin, okay, so that GFR go back to normal. So far clear? So far are we good? Yes, okay. Now students, look here. A comparison of the volume of filtrate formed per day is 180 liters. With that, urine released will be 1.5 liters. Okay. Suggested that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. Now you will be like, why are they secreting out if they have to reabsorb? Chumma, they can't make 1.5 liters of urine. Huh? They can't, no? This is what you thought, students. Have you ever thought like this when you are studying this chapter? Tell yes or no. Tell very, very honest answers I want. Honest answers I want, okay? You have to tell me honest answers. Come on, let me know your honest answers. Thought, huh? Chats are moving slow here, yeah. Yes, okay. Okay, if you are a student and if you don't think like that, then some problem is there, okay? If you're a student, you should think like that. Like how I used to think, why did apple fall on Newton's head? Like why didn't, like so many things were falling, no? Why didn't something else fall? I used to think like that when I was a child. Then now I realize that it makes so much sense because even urine is going out because of gravity, but then still muscles are there. I used to think so much about Newton at one point that, you know, it was like one addiction that I have to think about Newton every day. Why did he make gravity? Because I was not understanding physics. Okay, I used to only like the third law. Every action has an equal and opposite reaction. That's the only thing I wanted to learn. So yeah, happens, no? Happens, no? As a student. So many things. Only apple fell. I was feeling bad. If Shreyas sir listens to this, he's going to throw me out of the channel. Yes or no, students? Let's keep it very personal. Exactly, Sharad. That was my thought. So many things out of that one kutti apple fell. Right? Yeah. Okay, so students, secret. So many secrets we have, no? Yes. So this process is called reabsorption. Why do you make it if you have to reabsorb is the question. Now, but we have to study, right? At the end of the day, we have to study. Now, the tubular epithelial cells in different segments of the nephron perform this is neither by active or passive mechanism. Active and passive means what? By the cat, killed by the rat. No, that is what active and passive voice is, no? The cat was killed by the rat was killed by the cat. Students, remember? Yes, I'll give you a break. Even I'm hungry, yaar. You should think about me also. Even I'm hungry. I don't know even where to go eat lunch also. Yes. When we'll take? 2:30 we'll take. We'll just finish this urine formation, take a lunch break and come back. After that, only little is there. RAS mechanism, angio, this one, arterial mechanism and then little diseases. Then we are done with the chapter. Yes? Yes, very good. Active is by using energy, that means ATP is used. Passive means no ATP. Passive means no ATP. Okay. Now, for example, substances like glucose, amino acids, sodium, etc. needs energy to be reabsorbed. Okay. And reabsorbed actively. Who and all? Glucose, amino acids, sodium. Okay. Now, the nitrogenous waste are absorbed by passive transfer. That's why I told low threshold. Urea, uric acid is absorbed by no use of energy. That is because we need very less amount, students. So, when we need less amount, why to invest lot of energy, right? Yes. Now, I will not invest this much of energy teaching living world. Why? Because it will have only one question. For 4 marks, why will I put so much energy? So, even I will tell you people, last minute we will do one quick revision, I will tell you, right? But this chapter has 16 marks. So, I will put all my energy. And like if there are 11 students, then more energy I will put. So, that's the thing. So, whereas the nitrogenous waste are absorbed by passive transport, reabsorption of water also occurs passively in initial segments of the nephron. What are they telling? Even H2O will get actively and passively absorbed. It will be absorbed actively in the, that means in your LOH, okay, and it will be absorbed passively in your PCT. Even PCT, even in ascending loop also, sometimes water will be reabsorbed passively. So, but then they are telling, understood, for some period of time water will be absorbed passively. During urine formation, the tubular cells secrete substances like H plus and K plus and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps to maintain the ionic balance, okay, very important, and the acid base balance. That is the reason why H plus, K plus, ammonia, all this is released. You know, no, base, acid, base and acid, you know this, no? I am not going in depth, then you will forget chemistry. Okay, yes. So students, look here, let's see what and all is absorbed by each of them, okay? PCT. Now, PCT is lined by simple cuboidal brush bordered epithelium. This was a PYQ question. Okay, this was a PYQ question. Please make a note. Please make a note. Direct question came. They asked simple cuboidal epithelium, simple cuboidal brush border epithelium, like that, simple squamous epithelium. So they asked this direct question. So this was a PYQ. Now, which will increase the surface area for reabsorption. Okay, they are convoluted. That means they will already increase the reabsorption. Now, students, look here. Nearly all essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. Okay, by this segment, 70 to 80% water and nutrients are reabsorbed in the PCT. Okay. Now, PCT also helps to maintain the pH and the ionic balance of the body fluids by selective secretion of who and all? Nitrogen ions, ammonia and potassium ions into the filtrate and absorption of HCO3. So, who and all are there, students? Here H plus, K plus, NH4, then HCO3. Yes, this many people are going out in the PCT. Now, loop of Henle. Loop of Henle, who and all are going? Reabsorption is minimum in its ascending loop. Minimum. Okay, it's very less. Again, could be asked as a PYQ. In which loop? It is minimum in the ascending lymph. Okay. Now, however, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. This again you will understand. Medullary interstitial fluid. Now, the descending lymph of the loop of Henle is permeable to water but almost impermeable to electrolyte. This I have already made you write, so do not worry. This concentrates the filtrate as it moves down. Okay, understood? So, first students, here water will be given out, right? As it goes down and down, what is happening? So, here only water is getting absorbed. So, you get more concentrated and concentrated. Here it will become more diluted because water is still there. So, here students, important, the concentrate filtrate as it moves down, it will become very concentrated. Okay, concentrated. Now, the ascending lymph is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as concentrated filtrate pass upwards, it gets diluted. Is this clear to all my students? Is this clear to all of you? Tell me in the chat box. Yeah, brush border. No, simple squamous epithelium brush border. The entire thing was given as a PYQ. Not just brush border. Students, is this clear? How it is getting diluted and concentrated? Okay, here it is getting concentrated because all the water is getting absorbed. Here it is getting diluted because all the NaCl is getting absorbed. Simple. Wherever electrolyte is getting absorbed, dilute. Wherever water is getting absorbed, concentrated. Okay. Now, actively or passively, therefore, as a concentrated filtrate pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Okay. This you will study again in the countercurrent mechanism. Distal convoluted tubule, that is conditional reabsorption of sodium and water takes place in this segment. Okay, DCT is also capable of reabsorption of HCO3. Okay, DCT is also capable for reabsorption of HCO3 and selective secretion of, important, selective secretion of hydrogen and potassium ions. I would request all of you to just study the diagram in the NCERT where they are showing what is going out. No, just learn that. That is more than enough for you. Okay, and to maintain the pH. to maintain the pH. Now last paragraph students, this long duct, collecting duct, this long duct extends from the cortex of the medulla to the inner part, cortex of the kidney to the inner part of the medulla. Large amount of water could be reabsorbed from this region. Now very important thing is conditional reabsorption happens, right? Conditional reabsorption happens. There we have mechanism who ADH will reabsorb water and your aldosterone will reabsorb sodium. Correct? So DCT and collecting duct in normal condition, they will not reabsorb anything. They will only do during conditional reabsorption, they will do. Stress condition. So students could be reabsorbed from this region to produce concentrated urine. This segment allowed passage of small amounts of urea into the medullary interstitium. Again, where will you study this? In countercurrent mechanism to keep up the osmolarity. It also plays a role in maintenance of the pH and ionic balance. So, very important thing, DCT and your collecting duct will do only conditional reabsorption. Clear? Conditional reabsorption. Is this clear? Yes, okay, countercurrent mechanism, you will learn about this medullary interstitium. Shall we do one more question? Shall we do question and then we'll take a break, lunch break we'll take. Lunch break, wow, I feel like I'm in school. Shall we do a MCQ? Students, 1 second. Yes? Okay, I'll give the question. Ready? Yes, now I can see. Yes. Okay, students, first question. Can you see? Which of the following statement is correct? Which of the following statement is correct? Come on, answer all of you. Yeah, that is okay. Captos live is okay. That is, you can watch it after the class. Yes? One second, students. Chats are not loading. Yes, what's the answer? 1. Let's see the answer. Yes, perfect. 1 is the answer. 1 is the answer. Perfect. Yes? Yes, done. Okay, next question. I think one more question is there. 1 second. Yes, one more question is there. Students, in mammals, which blood vessel would normally carry the largest amount of urea? Think properly, which will carry the largest amount of urea? Can you all tell the answer? Yes, in mammals, the blood vessel which would normally carry large amount of urea. Two. Okay. Yes, yes. Correct, correct. Yes. Very good. Hepatic vein is the one that carries most large amount of urea. Okay. Large amount of urea. Good job. Now, how much marks did you get? 9 plus 4 plus 4. Please do the maths. Okay. This out of 17. Now your exam has happened. So put your scores out of 17. Yes, students, shall we take a lunch break? Shall we take a break to have lunch? When I'll go grab something and come very quickly. Yes, it is 2:32. Okay, we will be resuming. 2:32, no? Okay, we will resume back at how many minutes you want, students? Come on, quickly tell me how many minutes you want. Break. Quick, quick, quick. Yes, all of us are hungry. Let's go eat something and come back. If most of you are not eating, or if you already had lunch while watching my class, please write the notes of how much you have missed. Okay, but we all should eat lunch, so we will just eat and come fast. How many minutes, students? 20 minutes? Okay, so we will come back around 2:55. Okay, can be up to 3 p.m. Okay, I have to go down and come up. Yes, 20 minutes? Sorted? Okay, let's run. Okay, all of you join back. Okay, do not forget. Hello students, are you all back? Yes, slowly all of you are coming. Hello. Yes. Okay, we'll continue. Okay, students, so next topic is counter current mechanism. Okay. So by the time I draw, I hope all of you will join. Okay. Let me just draw. One sec. Yes, you can also draw. Those who join back, please do draw. Yes. Students ready? Yes. Yes. Yeah, Ramya, not bad. Not bad, not bad. Look at their observation skills. Doctors, no, definitely observation skills will be more. Okay. Shall we start? Okay, so students, this is counter current mechanism. Now, what is counter current mechanism? It's basically two tubes which are in close proximity. That means they're very close to each other, right? It could be blood vessels, or it could be loop of Henle, or it could be PCT or DCT. If they are very close to each other, in one of the tubes, if the fluid is going one way, in the other tube it would go the opposite direction. So that means the tubes which are in close proximity, the direction of the fluid will be different. Okay, vice versa. Clear? So here, thank you. Thank you, Vijayan. Okay, fine. Yes, students. So I'm getting disturbed because of your messages. What is this? These girls, yeah, the girls, it seems. Full gang and all they have made, the girls. Okay, students, let's continue. So I have basically drawn here, I've drawn loop of Henle and I've drawn vasa recta. Or you can normally say blood vessels, okay. So this one is the descending loop of Henle and this is the ascending loop of Henle, correct? Descending and ascending. Okay, this one, this is vasa recta. So this is the descending vasa recta and this is the ascending vasa recta. So students, counter current mechanism means basically if two tubes are in close proximity, the direction of fluids that flow in them will be opposite. That's all. That's all. Now look here. So imagine first the filtrate will be moving in the loop of Henle, right? So the filtrate will be moving this way. The blood will be moving in the opposite direction. Yes or no? Blood will be moving in the opposite direction. All of you agree? If filtrate is moving like this, blood will be moving in the opposite direction. So, now what is happening is counter current mechanism, one counter current, three counter current mechanisms are there. First one is between ascending vasa recta and descending loop of Henle. Can you see? Opposite direction. First counter current mechanism. Now, next counter current mechanism. Again, here the liquid will be flowing how? It will be going like this. It will be going like this. So that time the blood will be moving how, students? It will be moving like this, right? So opposite. Opposite direction. Simple. Second counter current mechanism. Okay. First counter current mechanism, second counter current mechanism. Then you will be like, ma'am, where is the third counter current mechanism? Simple. Look at DOH and AOH. They themselves have, the fluids are moving in opposite direction. Yes or no? If this is going downwards, this is going upwards. Okay, same way. If this is going upwards, this is going downwards. So there is one counter current mechanism between each of the fluids itself. Okay? Okay? Yes, done. This much is clear, students. This much is clear. Now, one very speciality about counter current mechanism is it does not happen in normal condition. It happens in stress condition. Now, my question to all of you. Which nephron will work during the stress condition? Is it going to be cortical nephron or is it going to be juxtamedullary nephron? All of you tell me in the chat box quickly. By then, I'll write the full form of all of this in one side. So, it will be helpful if you're taking these notes and studying in the last minute. By then, all of you tell me which nephron is going to work. Yes. So, AOH, DOH, AVR, DVR. Very good. Juxtamedullary nephron. Yes. So, yes. So, students, this works only in the stress condition, right? This only works in juxtamedullary nephron. So, basically what is happening, you know, basically we have to, do you see the space in between? Do you see the space in between? This space is your medullary interstitium. What is the space? Medullary interstitium. Now, in the medullary interstitium, I have to increase the concentration of two people. Who are those two people? Sodium and urea. Sodium chloride, that is your NaCl. Okay, I have to increase the concentration of NaCl and urea. That is the work. Okay, this is to maintain the osmolarity and to make sure that this person is already in stress condition. Now, no more water should go from this person's body. For that, I have to do extra reabsorption. That is why counter current mechanism is useful. Now, all of you know that your ascending loop of Henle is going to give out sodium chloride. Any doubts in this? It is going to give out sodium chloride. That sodium chloride will be taken in. This sodium chloride will be taken in by your that is vasa recta, and it will pass all the way. It will bring all the way till the ascending vasa recta and emit it or give out in the medullary interstitium. Is this clear to all of you? Is this clear to all of you? So, NaCl was given out by the ascending loop of Henle. Okay. I have to write LOH, no? ALOH. ALOH. DLOH. Students, 1 second. ALOH. DLOH. 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 Yes. Okay. Students, clear? Yes, those come and go, but we are permanent. Done? Okay. Okay, so these two people. So students, NaCl is given out by your ALOH. That will be taken in by your vasa recta and bring it all the way up to the top and drop it in the medullary interstitium. Now, NaCl concentration has increased. Now, one more person that we have job is with is your urea. So, this is your collecting duct. Okay, this is your collecting duct. Now, this collecting duct, okay, this collecting duct. Yes, okay. Okay, students, so this is the collecting duct. This will give out urea. This I mentioned in the starting of the class. Why? To maintain osmoregulation. Now you will understand what is that. Now that urea will be taken into the medullary interstitium. Now urea and sodium concentration has become increased in the medullary interstitium. That automatically what will happen? Water will start coming out of the tubules. Who will start coming out? Water will start coming out of the tubules. Is this clear? Okay, is this clear? Yes, okay, done. So, this is how they do maximum reabsorption of water during the stress condition. Very important mechanism, I hope it is clear. Okay, do you want me to quickly revise? We will quickly revise. So, there is two tubes. So, countercurrent mechanism means two tubes which are in close proximity. They are fluids which pass through them will be in opposite direction. Same fluids need not pass, different fluids. See, in this blood is passing, in this filtrate is passing. So, it can be different. So, when they pass, what happens? They are passed in opposite direction. Countercurrent mechanism happens in juxtamedullary nephron during the stress condition. What is basically happening is we have to increase the concentration of sodium chloride and urea in this medullary interstitium. How do we do that? We know that ascending loop of Henle will give out NaCl. That will be taken up by the vasa recta and drop it in the medullary interstitium. Same way, collecting duct will give urea, right? Collecting duct will give urea. Collecting duct will give urea, and that urea will be absorbed and dropped into the medullary interstitium. Clear, students? Yes. Oh, so many ads, yeah. Okay, that will also be given into the medullary interstitium. Now, the sodium chloride and urea will increase so much that the water, that means basically osmosis, students, from higher concentration to lower concentration, right? So, water will start moving from a region of lower concentration to higher concentration, and water concentration will increase, basically providing. So, the urine will become what? Urine will become more diluted. We will get more water out of it. Clear? So, urine will become more concentrated because we absorbed all the water from it. Is this clear? So, this is the reason how in stress condition our body works. Okay, done. Now, let's go to the next topic. Any doubts in this? Please feel free to ask. Okay. Now, students, understand, a person who has eats a lot of non-veg. Okay, a person who eats a lot of non-veg, what do? They have to drink a lot of water. Why they have to drink a lot of water? Because when the protein intake is more, okay, amino acids, urea intake is more, but concentration will keep increasing, no? So, that problem is urine will become way too concentrated. So, that's why a person who eats a lot of meat has to drink, that means a lot of proteins, has to drink a lot of water. Okay, H2O consumption, that means water consumption should be extremely high. And I had asked a question before this before this class, that which is the vein that will carry the most amount of urea? Answer was hepatic vein. I hope all of you understood that and answered. Yes? Yes, fine. Yes, countercurrent done and dusted? Yes. For my own life, they are telling me, they are not showing my own face to me. So bad. Okay. Yes. So many colors I have used. Done, students. Next one we will go. Okay. Okay. Now, hormonal regulation in kidney. We are going to come to our mechanisms. After mechanism, then we are done. Okay. We have only chuttu-muttu portions left that we will be doing now. Okay. Students, yes. First mechanism. RAS hai. Okay. One beautiful name. RAS. Very easy mechanism also. What is it? RAS. If it was some Hindi heroine, she would look so beautiful, no? Because the name sounds so beautiful. Now, all of you tell me RAS full form. Renin, angiotensin, aldosterone. Renin, angiotensin, aldosterone, right? Renin, angiotensin, aldosterone system. Yes? This is our RAS. Now, for RAS, yes, very good. Renin, angiotensin, aldosterone mechanisms. Very simple, students. We all understood juxtaglomerular apparatus, yes or no? Any student in my class who did not understand juxtaglomerular apparatus? Oh, stop. Students, something and all happened. So smart of me. Where am I? One second, students. I have to fix the problems that I make. Where will this come? Oh. One second, students. The problems I make are so funny that I can't even find solution for it. Yes. Okay. Finally, we reached here. Students, we understood juxtaglomerular apparatus. Yes or no? Yes or no? Fine. So, this juxtaglomerular apparatus, we understood about JG cells, right? Now, we will use that JG cells. JG cells was ready to release renin, right? I told you that renin will be used for RAS mechanism. No, now we are going to see RAS mechanism. Slowly, we have to do complicated. Yes. Final destination achieved. Okay, students. So, RAS mechanism. Exactly. Life full of problems. Upon that board also problems. Okay, students. So, juxtaglomerular apparatus. Right. Just juxtaglomerular apparatus was used to give us renin. Okay. This is pakka clear for us. Is there anyone in my class who did not understand? Okay. It gave us renin. Now, I told you this renin was released because the filtrate had some problem. Now, what could be the problem? Problem can be like, see, like how many problems can come? Problems are not less, students. Solutions are less. Problems are more than enough. So, students, problems are BP becomes low, okay, or H2O is less. I'm telling these could be the problems that is there in the filtrate, okay, water. So, I shouldn't write water, I should write blood volume. So, it is because terminologies are important, okay. Blood volume is low. Yes, this could be the reason. BP can be low, blood can be low, blood volume can be low, even that is the, I forgot. Okay, glomerular filtrate can be low. Glomerular filtrate can be low. Yes. Okay. And then, students, I'm sorry for. Yes. Okay. No, this is recorded class 3. This is recorded class. Okay. That's why I'm able to see your live message. It's recorded. This is live, dear. He's telling yes, class is live, it seems. Yes, tongue is twisting, students. Okay, so this is low. Okay, again if I tell it will twist again. So then students, blood, Na, sodium and Cl will be low. That means salt, if salt is low, then BP also will be low. So these are the problems. So we don't have less, so we have more problems. Okay, okay. Yes. Now renin was released. Renin was released with so much telling that yes, I will solve all the problems in the life. But Papa, it got failed, students. What happened is renin came. Okay, renin came telling that I will try to solve as much as problems possible. But now what is going to happen is this renin will be released into the blood. All of you know hormones will be released into the blood. Endocrine we did. So this is the blood vessel, sorry, this is the blood vessel. Now in the blood vessel there is a protein called as angiotensinogen. Who is there? Angiotensinogen. Angiotensinogen is there. Whenever tensinogen, okay. Did I say BP also glow? Oh no. Did I say glow, students, instead of low? Don't worry, at least that is glowing. Skin is not glowing, life is not glowing, but GFR is glowing, BP is glowing. Oh my god, if students who watch this live recorded, they'll be like, what is wrong with ma'am? For her tongue is tossed. Students, maximum 3 hours my tongue will stop, like it will not twist. After the third 3.5 hours, then my tongue is like, okay. Yes. Okay, okay, sorry, I told glowing. Okay, students, listen, angiotensinogen is an inactive form, okay, inactive form, which is there in the blood plasma. Okay, this is an inactive form, and where is it present? In the blood plasma. Now, renin has come to activate this. Renin has come to activate this. Actually, this angiotensinogen is released by the liver. No doubt in that, no, it is released by the liver. Now, renin has come, students, but renin is not able to completely activate this angiotensinogen. So, with the help of renin, we will form angiotensin 1. What will form? Angiotensin 1. That means it could change to one level, but it could not change completely. Is this clear? Okay, is this clear? Yes, angiotensin 1. Okay, yes. Now, this angiotensin 1 will not be enough for us. We need one more person called as angiotensin 2. But for that, we need someone's help. Whose help we need? We need the help of lungs. Okay, we need the help of lungs. Lungs will release something called as ACE. Lungs will release something called as ACE. Can all of you tell me what is ACE? ACE, very good. Angiotensin converting enzyme. Yeah, correct. When you speak bio for 4 hours straight. Okay. Students, what is ACE? Angiotensin converting enzyme. So, what is it supposed to do? It is supposed to convert angiotensin 1 to angiotensin 2. Easy, easy. Angiotensin 2. Now, angiotensin 2 is the real hero. Angiotensin 2 is the real hero. Yes, very good. Angiotensin converting enzyme will convert angiotensin 1 to angiotensin 2. Okay. So, we have taken different people's help. Okay. So, first we took liver's help, this was in the plasma. Okay, I'll write this. This is present in the plasma and it is a protein. Okay, this is a protein. Now, angiotensinogen was converted with the help of renin. Okay, with the help of renin to angiotensin 1. From angiotensin 1, we had to convert it to angiotensin 2. We did not have anything else. So, we took the help of lungs to release ACE, which is angiotensin converting enzyme. This person helped in converting, okay, angiotensin converting enzyme helped in converting angiotensin 1 to angiotensin 2. Now, what is all this? Why are we learning all this conversion? We are learning all this conversion to solve all these problems, right? To solve all these problems. To fix all the problems, we have brought a hero into our life, right, students? So, the hero is going to do what and all? Let's see. Let's see what and all hero will do. First, we'll give one problem for the hero student, then he'll leave and go. So, first problem we'll tell. What is the first problem? We have BP is low. Okay, my BP is low. So, we will tell what happened to my mic. What happened to my mic? Is it not proper? Hey, chumma people don't tell. Oh yeah, some problem is there, no? It's fine, no? Wait 1 second, students. Is it better now? The voice better now? Is it better now? Tell me all of you. Yeah, the mic is too long, that's why. I'm getting caught inside, stuck inside the mic. Wait, let me see. I'm not talking, huh? Is it better now? Yeah, it's better, no? Student, the mic is, the wire is too long. So that's the reason. Okay, fine now? Was it there for a longer time or was it there for a less time? Little time only, no? That problem came. Okay, yes, fine. So students, angiotensin 2, we will give one problem. What is the first problem we have? The first problem is that our BP is low. If BP is low, that means we have to increase the sodium, salt content in the blood. So what we will tell? We will tell one thing, you do one thing, you stimulate. This angiotensin 2 will stimulate adrenal cortex. Who it will stimulate? Adrenal cortex to help in reabsorption. To help in, so it will help, it will ask adrenal cortex to release aldosterone, which will actually stimulate. Okay. Okay. Since lunch break, okay. So students, it will tell adrenal cortex to release aldosterone. Okay. This aldosterone will send signal telling that do conditional reabsorption. Sterone, which will do conditional reabsorption of what? It will do conditional reabsorption of sodium. Simple. First problem sorted. If conditional reabsorption of sodium happens, okay? Oh, is it? Okay. Okay, understood. Okay, now it's better, I feel. Okay, students, so conditional reabsorption of sodium will happen. Now what is going to happen? When sodium amount increases, obviously BP also will be increasing. No? So first problem sorted. Is this clear to all of you? Clear to all of you? Yes? Okay, done. Now second problem we'll give, okay? Second problem is that our blood volume is low or GFR is low. So the second problem will be sorted. Second problem will be sorted by, okay, second problem has to be sorted by increasing the GFR, okay. For increasing the GFR, what we should do? We should do vasoconstriction of the efferent arterioles, okay. More efferent, from efferent more filtrate should come, okay. So for that, we will do vasoconstriction. Vasoconstriction, okay. Of whom we'll do? Not of efferent, we'll do efferent, okay, so that the filtrate does not go, right? The filtrate, more filtrate comes in and less filtrate goes. So that is vasoconstriction. This way what will happen, students? Automatically blood volume will increase and the GFR will increase. Two problems sorted. Two problems sorted, okay. I'll write here in bracket EA. Yes. Done. Second problem solved. Is it too confusing? Should I reduce the size of the problems? I've given too much importance for the problems, no? Wait, students, I'll write it small. Otherwise, it looks too congested. 1 second. Okay. Now, I think it will be more clear. Students, once again, we'll write the problems this side. Okay, we'll write the problems this side. So, what is the problems? Which problem did we solve now? We solved the problem of BP low. BP was low. Now, GFR also we reduced. GFR was low. Automatically, blood volume will be low. blood volume will be low, and then H2O was less. Okay, the level of H2 also was less. Now in this, how many problems we sorted? We sorted 1, 2, 3 problems cleared. Done? Okay, now it's more spacious, no? Now you can see clearly. Yes, fine. Okay, vasoconstriction, done. Now students, next one is what? The next problem is water is less. Okay, water is less in the blood. So when water is less, the only thing we have to do is we have to allow reabsorption of water. So we will tell reabsorption. We will send signal for reabsorption of H2O. Got it, all of you? So solution done. Okay, solution done. Is this clear? Okay, is this clear? Yes. Done, done, done. Angiotensin 2 done. Okay. Okay, done. Very good. So students, RAS mechanism perfectly alright. Perfectly alright. I am not repeating it, okay, because I think I explained it really good. So you don't have to, but is there any, like you want any of it to be written properly, just let me know, okay. For your notes purpose, I am telling, okay, for your notes purpose. Yes, done? Okay. Next students, next one will go, that is your ADH. Okay, next one is ADH. Yes. Next is ADH. Anyone knows the full form of ADH? Anyone knows the full form of ADH? Come on, tell me in the chat box. Anti-diuretic hormone, right? Anti-diuretic hormone. Students, what is happening here? What is happening here? Basically, loss of water, right? So, against the loss of water, we have a mechanism. Okay, diuresis is a condition which comes for people who have diabetics. Like they will loss of water will happen, right? So anti-diuretic. Yes, very good. Anti-diuretic. Okay, students, listen. So this one, this anti-diuretic hormone is synthesized by your hypothalamus. Okay, and it will secreted out by your posterior pituitary. Right? Posterior pituitary. Got it? Posterior pituitary. Now students, understand what is basically happening. Okay? Yes, vasopressin. Now students, what is basically happening? Antidiuretic hormone, which is synthesized by the hypothalamus, secreted by the posterior or released by the posterior pituitary, they are actually regulating. So what is the problem? That is the problem is that osmolarity has changed of what? Blood. I told you blood osmolarity has to be 300, or it can be even, you know, less than 300 but not more than 300. So the problem is blood osmolarity has changed, or it has become more than 300. Okay, more than 300. Now during this condition, what is expected? What will happen? Salt is more, water is less. Now we have to increase the water content. Simple, simple. We have to increase the water content. Okay, yes, got it. Fine. Now for that to happen, first thing, it will actually tell us to drink water. First solution is given by activating the thirst center. Activating the thirst center, telling us to intake more water. That means exogenous intake. Outside say we have to drink water, no? So exogenous intake. Simple. Activate the thirst center, tell us to drink more water, exogenous intake of water. One way of intake of water has happened. Second one will be what, students? Obviously reabsorption. Other than that, what can we do? So next one, what they do is they will stimulate conditional reabsorption of water. Okay, whom will they tell? They'll tell the DCT and the collecting duct. They will tell the DCT and the collecting duct to reabsorb water, to do conditional reabsorption. To do conditional reabsorption. Is this clear? Very simple mechanism. Only RAS has so many terms to remember, but these are very simple. These are very simple. Okay, very good. So, students, what will happen at the end of this? The urine will be concentrated. Why? Because we are removing, trying to maintain the water. Like we don't want the water to go out. So, the urine formation will be concentrated. This way, we will save all the water that is lost. Okay, all the water that is lost. Simple, easy, very simple one, very simple one. Okay, very simple one. Next one, okay, next one, is this clear? Next one is ANF. Next one is ANF. ANF, can you guess the name? Atrial natriuretic factor. Okay, atrial natriuretic factor. Got it, all of you? Students, this is basically released from where? Released from the heart. Okay, this is basically released from the heart. Now, when this releases from the heart, what is happening is this is actually against the RAS mechanism. This is actually against the ADH mechanism. So, it is going to do, so two things are there. First thing, diuresis and natriuresis. So, what is RAS doing? It is increasing the sodium level. Right, it is increasing the sodium level, so BP will become normal. What is ADH doing? Absorbing the water, so the water loss is, not there. This fellow is doing opposite of both. This fellow is doing loss of water. Okay, what is this fellow doing? Here he is doing loss of water and here is doing removal of Na. So, he is against both of the mechanism. This is the way our body balances, students. This is not a bad thing. This is the way our body will balance. Is this clear? Yes, very good. Antagonist to your RAS, right? Antagonistic to your RAS and even ADH. Okay, because here loss of water is there, no? Diuresis, it's fighting against diuresis, no? So, it is against both the mechanism. Is this clear? So, students, what is happening? Why is this person doing this? Problem should be there, no? The problem is ulta of RAS. Whatever you studied in RAS, write the ulta. High BP, okay, high H2O in the water, like more diluted, okay, GFR is more, sodium content is more in the blood. This is the thing, ulta of RAS, ulta of RAS. At the same time, this only we can write high blood volume, high blood volume. Now, all this has come. So, for this to be counterfeit, this is the best mechanism. Okay, clear, students? Very good, very good. Yes, hypertension, okay? Yes. Yes, they will do vasodilation of who? Efferent arterioles, no? So, the more filtrate will go get filtered. Yes, students. So, the mechanism is not mentioned, just that it is antagonist to RAS and ADH, like it is opposite. It works opposite to RAS and ADH. So, this way our body will balance the osmolarity. Okay, our body will balance the osmolarity. Is this clear? Very easy, no? Very easy, no? Students, yes or no? Easy or no? Understood or no? Yes? Okay, understood. Great. Students, 3 mechanisms over. Any doubt in this? Any doubt in this? No, right? We are going to micturition, that's why. Next topic. Wow, yeah. I want to clap for all of you. I want to appreciate all of you because you people almost going to be for how many hours, yeah? 12 to 1, 1 to 2, 2 to 3. 2 to 3? Yes, almost going to be 4.5 hours. So I cannot tell you, I cannot clap for you people, but I'm telling you, I appreciate you people all stayed through from the starting till the end, which is shows how dedicated you are. And yes, you people will definitely do amazing in your life. Okay? Yes. I always nice when students stay back till the end of the session, not just come in the starting and go. Okay, that means basically wording of the urine, students. The word might look like very fancy, nothing is there. Oh, why is arrow coming? Something happened. Students, 1 second. Board is doing little dancing. Stay there. Okay, it basically means voiding. Oh, something happened. Board got fed up. Okay, can't do anything, students. Let's continue writing. Okay, students. So, video also got stuck. Oh no, what happened? Am I stuck? Okay, video is stuck, but you can hear, huh? Okay. 1 second, students. 1 second. Students, I'm alive. Yes. Yes, 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 yes. Yes, yes. What is your audio? I think now it's working. Yes, working? Yes. Okay. Since camera got tired. Camera got tired of me talking. Okay. So yes. Okay. So yes. What is the problem? Yes, we are finally back. Since I don't know what is happening, give me 1 minute. I will fix all the problems at once. I think it's because of this. Now it's fine. Yes, students, is the voice better? Mic also is issue. One second, one second, one second. Hello, is it fine? One second, let me check, let me check. Students, is it fine or should I just, why is it, hello, is it fine? Yeah, it's fine, right? Yeah, okay. So students, the camera got a little problematic, but I think voice is fine now, no? All clear? Okay, let's continue. Yes, thank God. Okay, students, so voiding, okay, board it's writing, right? So basically voiding of urine. Something is wrong with the board again. Okay, no problem. Let's continue. Let's, students, any other problem, please let me know, okay? Yes. Okay. So yes, voiding students, before everything shut down, so let's finish this chapter. Okay, so students, we have to understand the pathway of urine. Okay, the pathway of urine, that is basically, 1 minute, try another pen. Okay. Okay, no, it's not working. Huh, okay. Fine. Okay, so students, urine will go all the way to the collecting duct. Excuse me, sorry. Okay, go all the way to the collecting duct. What is wrong? Exactly. Okay, so collecting duct, actually, I think this studio, no one takes that long of classes. That's the reason. Like all 2 hours classes, no? That's why board is like, why is this lady torturing me so much? So, students, it will go to duct of Bellini. That means your column of Bellini. Okay, from that, it will go to, right? Is the problem? Problem is not there, no? Let me know, okay? I won't know otherwise. Am I still stuck? Yes. Students, 1 minute. Is there any problem? I cannot see your chats also. Yeah, last period, last class, it's happening. Yes, no problem. Okay, okay, fine, fine, fine. Okay. Students, so duct of Bellini is there. From duct of Bellini, where will it go? Minor calyx. From minor calyx, it will go to the major. From major, where did I tell you it will go? Pelvis, renal pelvis. From pelvis, where it will go, students? From pelvis, it will go to the ureter. Ureter, urinary bladder, outside. Okay? Ureter, urinary bladder. From urinary bladder, it is going to urethra. Urethra, it will be expelled out. This is the pathway of urine. Yes or no? This is the pathway of urine. Okay. So, your renal pelvis, okay, do not forget this is not just pelvis, your renal pelvis, okay. Renal pelvis. Yes. Okay. Now students, what is the reason for the yellow color of the urine? It is presence of a urochrome pigment, right? Presence of urochrome gives the yellow color to the urine. And what is the pH of urine? pH is around 6.6. 6 is the pH. Okay. And what is the amount of urea that is present in urine? Around 25 to 30 grams. This is how much urea will be there in your urine. Okay, clear? Simple. So students, here you already know, I told you in the starting itself, urinary bladder, what is happening? The detrusor muscle has to contract and the sphincter muscle has to relax, then only urine will come out. And how much of urine is going out? 1 to 1.5 liter of urine will be expelled out. Okay, expelled out. Done? That's all. Okay, that's all about your micturition. Okay. Okay. Yes. 1 second. Yes. Students, next topic, accessory structures. No, hilum, see, hilum is in that side, no? So, if I draw a kidney like this, hilum is here, right? This is your renal pelvis. Hilum is only for the arteries and veins to go. Okay, arteries and veins to come and for the nerves to move around. Okay, clear? Okay, students, accessory excretory organs means nothing but from where other parts is, you know, excretion happening, like it could be sweat, it could be urea, it could be anything, right? So, who are those people? Who are those people? We'll just quickly write it. It is first your sweat, because this board is not allowing me to write, so that's the reason. Okay, sweat, sebum. What else? Can all of you tell me one point? Sweat, sebum, saliva. Is this all excretory? Yes or no? Some amount of waste is coming out of all this. Then from lungs we give out carbon dioxide. Yes. And the last one is liver. This is mentioned in your textbook. One paragraph is there for you. All of you check there. Yes. Students, sweat, sebum, saliva, lungs, liver. Now, what is each of them giving? I'm going to write that. Okay, each of them is giving what? I'm going to write that. First one is NaCl will be given out. Salt is definitely given out. If you have ever, you know, your sweat has salt. Some amount of urea, some amount of lactic acid. Lactic acid. Okay. Yeah, the pointer is not leaving me. Pointer is not leaving anymore. So now I have to get used to this. Something I did. Yes. Yes. Students, let's not touch anything before this will collapse. Students, pointer when see. Wow, good to do some things, no? See, I found out the method. Yes, very good. Uric acid, lactic. Now here students, sterols are going, hydrocarbons are going, some amount of hydrocarbons are going and waxes are going. Who are all? Waxes. Now, saliva students, saliva a lot of things are not going, but mainly some amount of water will go. Okay, some amount of water will go. Okay. Yes. This is how Eureka, that means, you know, they try to do something else, they find out something else, no, that is exactly what happened. Okay. Yes, okay. Students, saliva, what is there? Some amount of water will go, very less amount of carbon dioxide will be removed. Now, lungs, all of you, sorry, why am I telling saliva, carbon dioxide? Lungs, students, lungs. Okay, lungs, some amount of carbon dioxide will go. Yes or no? Okay, very less amount of carbon dioxide. In lungs, actually, in your textbook, one thing is mentioned that 200 ml per minute of H2O is given out. Very important, very important. This is like a number, so PYQs could be asked. So just make a note of this. Okay, just make a note of this. Again, saliva with some water, nitrogenous waste. Students, water is very least amount. Okay, very least amount. Okay, now last is liver, bilirubin, cholesterol, steroid, all of this you know, right? Bilirubin, bad cholesterol, right? Cholesterol, all this will be given out with the help of your liver. Done, done? Yes. Now, students, artificial kidney, last topic. Then, I think disorders you have, disorders, wait, one second. Let me check if I have added. Yeah, disorders I have added, students. Disorders, yeah, disorders I have added. So, yes, last topic of our class is dialysis. Dialysis means what? If a person's kidney fails. Okay, if the person's kidneys fail, both the kidneys fail. You know that for us, one kidney say all the work will happen. But both the kidneys fail, what will we do? We will do an artificial kidney, that is dialysis. So in this, what is happening? The excess amount of waste will be removed out. Okay, excess amount of waste will be removed out. Okay, yes or no? So students, what is happening in artificial kidney? Okay, so only this much you have to know. There is a cellophane tape. There is a cellophane tape which will be put. Simple, it is given in your textbook. That only we will learn. This is the complicated one. Okay, cellophane tape is put, students, and there will be a dialyzing machine. In this machine, dialyzer will be there. Okay, who will be there? Dialyzer will be there, and in that dialyzing fluid, no urea will be there. So this way what will happen? Diffusion will happen. From a region of higher concentration to a lower concentration, urea will come out. Simple as that. So like that, urea will come out. Simple. So that is all mentioned in your textbook. So this dialyzing fluid will not have any urea. Okay, this dialyzing fluid will not have any urea. This way what will happen? From the blood, right? The blood which we took from a person, that blood, all the urea will start coming out from a region of higher concentration to a region of lower concentration. Clear or no? Clear or no? Clear? Okay, done. Fine. Students, now, look here. Here only thing is heparin pump to prevent clotting. This I told you. Heparin and anti-heparin. I told you in body fluids, heparin will be injected first so that the blood does not clot, because if the blood clots, the processes fail. Then at the end, before giving it back to the body, we'll put anti-heparin. Okay. That's the only thing that you need to know from this. Is this clear, students? Is this clear? Yes. Okay. Okay. Now, disorders of the excretory system. Now, what and all can happen? Okay, the first thing is, students, accumulation of urea. I told you, if urea, we need only less amount of urea for osmoregulation. But if that becomes more, right, that will cause uremia. That will cause uremia, which is highly harmful and may lead to kidney failure. Now, in such patients, urea can be removed by a process called as hemodialysis. This is called dialysis. That means artificial kidney. Blood drained from a convenient artery is pumped into a dialyzing unit after adding an anticoagulant like heparin. This my students already know. The unit contains a coiled cellophane tape surrounded by a fluid, that is the dialyzing fluid, having the same composition as that of the plasma, except urea, except the nitrogenous waste. Okay, the porous cellophane membrane of the tube allows the passage of molecule based on the concentration gradient. Simple diffusion. Simple diffusion. Okay. As the nitrogenous waste are absent in the dialyzing fluid, these substances freely move out. These substances freely move out, thereby clearing the blood. The cleared blood will be pumped back into the body, and that time what we will put? We will put anti-heparin. Okay. Students, this is a blessing for a lot of people, but at the same time, it's a problem. Okay. Now, last topic is Kidney transplantation is the ultimate solution. Dialyzing is very expensive, and it is not, however rich we are, we cannot do it, because every month doing it, or within 3 months, 6 months doing, depending on the, how much body is doing nitrogenous waste, it is not possible by a person to handle so much of financial crisis. Okay. Yes. Okay. Yes, done, students? Okay, fine. So here, next is your renal failure. So that when kidney transplantation is the ultimate solution, dialyzing is not an ultimate solution. Okay, a functioning kidney is used by a transplantation from a donor, perfectly a close relative, to minimize the chances of rejection by the immune system. Now here, what will we give? We will give cyclosporin A as an immunosuppressant for avoiding graft rejection. Clear? Clear? Now, renal calculi, stone or insoluble mass of crystallized salt formed within the kidney. Kidney stone, all of you know kidney stone, no? That is renal calculi. Okay. Now, glomeruli, students, what's happening? Are you understanding? Okay, yes. Glomerular, okay, glomerulonephritis is inflammation of the glomeruli of the kidney. Okay, that will also cause problem. Why GFR will be troubled. Okay, because it has to come in and go out, no? Okay, let's do quizzes very fast. Otherwise, now I have to give it to Diksha ma'am. Do quizzes very fast. Okay, and will you all comment after the class how the class was? Oh my God. Yes, students, class is done. Now answer the questions. Read the questions quickly. All PYQs, okay, previous year questions. Yes. Students, those who do not know gout, okay, that was there in the starting part. That means excess amount of uric acid in the body will cause gout. Okay. Very good, yeah. So fast. Let's see the answer. Yes, good job. 4 marks. So 17 was the marks. Now plus 4 you add, okay? Plus 4. So D is the answer. Very good. So which is the option? Glomerular nephritis is, no, A, no, sorry. This one, 4, B, 1. Okay, then students C, 2, D, 3. I don't know how to do maths the following, but you understood, no? Yes, next question. Which of the following causes an increase in the sodium reabsorption in distal convoluted tubule? Okay, distal convoluted tubule, which is the one? Come on. Diksha ma'am started her. No, she didn't start, no. She started? Oh no. Okay. Shreyas also is live. How many are live? Students have to end the class now. 1 second. Do the answers fast. Do it fast. So we can end it. Yes. Students, answer is 1. Okay, 1. Yes. So, yes, students, I will end the class because so many lives cannot happen. Okay, we will do this later. Okay. So, yes, thank you so much for joining. I don't know if I'm even in the class. Yes. Students, I'm going to end the session. I'm so sorry. Thank you so much for joining. It was a very long session. Those who stayed, I'm so happy for you people. Let me know in the comments how you felt about the class. The PYQs and the questions, I'll post it on the Telegram. Do join our Telegram channel. It is Vedantu NEET English. And you know that the crash course is launching tomorrow. So if you want to join, quickly join. The link is in the description. And do join our Telegram channel. That is Vedantu NEET English. Okay? Yes. Vedantu NEET English Telegram channel. You will get all this PDF there. Bye students, see you all. I'm sorry I have to end it very abruptly. But yeah, because so many people are going live in one channel. Yes, I'm so sorry I'll end the live. Bye students, thank you so much. Do not forget to put comments, okay? Do let me know in the comments.